Welcome everybody. Welcome everybody to this exciting and intense session of Chasing COVID. My name is Jordi Naval. I am the CEO of Biocat, and I will be your host this afternoon in this interesting and exciting session. This virtual workshop is made possible thanks to in collaboration with the Directorate General of Research and Innovation of the Department of Health of the Generalitat de Catalonia, also Directorate General for Research of the Department of Business and Knowledge, and La Caixa Foundation and EIT Health. So during this event, we will have 4 blocks of 5-minute presentations. It's going to be 5 presentations on each of the 4 blocks. And our aim is to have an overview of the main and most advanced research projects from our whole ecosystem related with COVID research. We will have a first panel about reprofiling, a panel about vaccines, a panel about new epidemiological studies, and a panel, I said everything. I mean, repurposing new therapies, vaccines, and genetic studies. So the goal is that, and to make things a little bit more interesting, we have decided to vote for the best projects. We have 20 projects today, and we have decided to vote, for you, for the audience, to vote for the best projects in 2 categories. The first category is beautiful science. So I invite you, all the attendees today, to vote for the project which, in your opinion, is more attractive in scientific terms. You recognize beautiful science when you see it, right? So this is the first category. The second category of the voting will be a game changer. Those projects that really can have a practical application and can be deployed in the following months to help us cope with the COVID pandemic. So these 2 categories, the winner out of the 20 projects, the winner in each category will receive a prize of 1,000 euros. It's not that much, but I think it's a recognition of all the effort that you have been deploying over the recent months. And also, very importantly, this voting, bear with me. It seems complicated. It's not that complicated. Just it's quite simple. 4 blocks, 5 projects, each block, 5 minutes presentation per project. At the end of each block, we will have the voting of the 5 presentations we just saw, okay? And this, we will repeat that over the 4 blocks during the session, right? And so we will have the winner at the end. To make things a little bit still more interesting for you, and because today we have more than 300 registered people, registered scientists in this seminar, in this open poster session, to make things a little bit still more interesting, we would also vote in parallel, starting now. We will also vote in parallel which is going to be the first vaccine approved by the European Medicines Agency. It's going to be Oxford. It's going to be the Oxford team with this chimp adenovirus vector. It's going to be CanSino team with the adeno5. It's going to be Johnson & Johnson with adeno26. It's going to be Moderna or BioNTech with their RNA vaccines is going to be maybe CanSino. I mentioned CanSino. It's going to be the Sinovac Sinopharm. I mean, the Chinese company with a classical inactivated virus. So you see on your screen the voting. I see already the Oxford team is winning the race. So let's see what happens. This is going to be amazing to see the results because you are the experts. So it's very important. I think it's very important for everybody that we have an opinion. I know you have your favorites. I have mine too. So at the end of the presentation, at the end, like around 5:30, more or less, we will have the final voting, and we will have Dr. José Luis Cabero from ILX Therapeutics commenting on the final result. It's like after the Barça match, you have a postpartit. So we'll have the postpartit and we will comment on the final results. So all the panelists can vote too. Everybody can vote. So please, let's get actually started. Close all your other Zooms, Teams, phone calls. This is, I hope it's going to be a very exciting afternoon. And thanks for all the panelists. Thanks everybody for sharing your projects with you. So let's get started. The first panel is going to be about repurposing. It's about using drugs that have been approved for other indications, the use of these drugs specifically in dealing with a COVID infection or with the COVID secondary effects. The chair of this panel will be Dr. Ana Massagué. Ana, hello, how are you? I mean, from the Directorate General of Research and Innovation of the Department of Health. So, Ana, the floor is yours.
Thank you, Jordi. Thank you for inviting me to share this session. I mean, one of you.
No, come close to the microphone.
Okay. Now?
Better.
Okay. So thanks, Jordi, for inviting me to share this session, which, as you said, is about of drugs, animal models, where to. The speakers from this session are Maria José Buzón and Alejandro Genescan from the group of infectious diseases of the BioRegion Institute of Research, Baldomero Oliva from Structural Bioinformatics Group of the Fabra University, Josep Per and Andrés Anton from the Liver Unit and the Respiratory Viruses Unit of the BioRegion Institute of Research, Joaquim Segalés from Cresa Atirta, and Alejandro Peralta from GPCR Drug Discovery Group of the UNIM, led by Joaquim Segalés. As Jordi said, you have 5 minutes each to go with your talk. At the end, we'll have 5–10 minutes of discussion. And something that I was told, but I don't know, Jordi, you didn't say that, but according to the instructions, you need to vote for the beautiful science or the game changer during discussion. Because after discussion, the section is closed and we're going to vote anymore. Okay? So please, Maria José, if you are so kind, you can start. Thanks.
Thank you, Ana, for the introduction. Do you hear me? Yes? Okay. I need to see my slides. Okay. So this work has been performed primarily in the BioRegion Research Institute, and this is the team members and the collaborators. And the main goal was to establish a physiological model for the study of viral entry and also the testing of antiviral drugs against SARS-CoV-2. And this is because current methods are using cell lines mainly, but they have intrinsic limitations, as for example, the differential expression of host proteins that are involved in the viral entry. So the first thing that we did, because based on that, we want to establish a new model, but in this case, using human explant tissues using lungs. So the first thing that we did was to generate pseudoviral particles because it allows us to work in a VR2 facility. So we obtained the expression of the spike protein from the SARS-CoV-2, and we use the backbone of the BSV-G virus to construct our pseudotypes. And those pseudotypes, they express the luciferase gene or the GFP. And then, so we can see in the right graph that the titer of the virus are pretty high and pretty good. So in the next graph, this is an infection using Vero cell lines that are cells that are very susceptible to get infected with this virus. So the infection that can be blocked very nicely using an anti-AC2 antibody, because the AC2 is the main receptor of the virus for viral entry. And in the next graphs, this is an example of antiviral testing of different drugs using the cell line. So for example, we have some antivirals as the one shown in green, that they are potential, they have effect on the cell line. Some others in orange that they have some effect, but at very high concentrations. In here, we are showing 100 micromolar. And even we are able to see some drugs that are increasing the viral entry within the cells. So next slide, please. But as I said before, we wanted to establish a more physiological model. So we took human lung resections from surgeries, and then we disaggregate the cells and we perform the ex vivo infection assays. And in the next graph, you can see here an example of a flow cytometry plot of the cells. In here, you can see that there is a expression of the AC2 receptors in lung cells, and also those lung cells are able to get infected using BSVG GFP in this case. So we see around like 24% of infection. Also using this model and adding the antibody anti-AC2, we can block the infection in lung cells in a dose-dependent manner. But in the right graph, you can see different examples of different patients, and this blocking effect is somehow different depending on the donor that we are using. So in the next slide, please. So here is the parallel testing of some antivirals using the both models, the Vero cells and our lung tissue models. And these are some examples that we have testing. And for example, the first drug is Camostat. So Camostat, as you might know, is an inhibitor of one protease, a cellular protease that is required for viral infection, but this cell line does not express the protein. So no antiviral effect is observed. However, in our lung tissue model, we see a very nice antiviral effect. The second drug is a drug that is able actually to work pretty good in both models. However, if you look at the IC50, is somehow a bit higher, like 1 log higher in lungs compared to the cell model. And the third drug is, antiviral drug also it doesn't work in cell lines, but it's working somehow in our lung cells. And the last example is a drug which is enhancing like threefold the infection in beta cells, but no effect at all and not increasing the infection when using real cells from human lungs. So the next, so cell lines do not always mimic antiviral testing in our tissue lung explant model. This model is relevant and more physiological for the screening of potential antivirals and to study SARS-CoV-2 entry mechanisms. What we are doing now, so we are continuing testing potential antivirals. We have close to 40 right now. We are also working on the identification of alternative cellular targets for SARS-CoV-2 entry in the cells, in lungs. And also we are working on evaluating the influence of the immune microenvironment on viral entry and also the efficacy of antivirals. So thank you so much for your attention.
Can you hear me? Yeah?
Yes.
Okay, thank you very much, Anna, for the introduction. This is a work published recently in PLOS ONE. It's an in silico drug repurposing combination of several drugs, and you can see that there are several institutions and groups that have been involved, public and private, for this work. So in the next slide, I'm going to introduce the methods that we have used. On the one side, we have a program developed by Anaxomics. It's called TPMS. Is based on neural networks, artificial intelligence approach, and neural networks use a network of connections, of nodes, and then with an input and an output. In the input, there is the information that comes to the system, and in the output is for the training of it. Then the other method that we are using is Gildify. Gildify in the right side, it's a method developed in our group. This is based on PageRank algorithm, the algorithm that has been used by Google for when you are searching for web services. And it uses a message passing. It transfers information from one node to the other, and it covers the whole network. For the network, we use the protein-protein interaction network in both cases. In the next slide, please, we can see what are the characteristics that are common for these both methods. And this is the use of an input, that is our gene disease associations or drug target associations. Then all the nodes get some kind of a scoring from the system, and that is going to tell how relevant are these nodes or proteins for the pathology that we are going to study. And on this, we can get some kind of pathways with the relevant nodes that connect from the input to the output, the result. There is a difference between both methods. TPMS uses a result, an objective defined by the nodes that is going to receive the answer from the input, while Gildify doesn't have this output. This means that the system transfers all the information along the network, and then we have to use a threshold to get just the top ranking ones. Then we can use these both approaches to study the potential application of drug combinations. So we have a set of nodes associated with a disease in yellow that makes a network, and then we can have one or two or several drugs, and what is the influence of these drugs through the network, transferring the information to the pathology. So that's what we have used for SARS-CoV-2 infection. And in this particular case, what we have is the network is just the interactome network, all the protein-protein interactions, then the input. For the input, we have all those targets that we know have been involved in the infection, such as ACE2 or TMPRSS2 or furin. These are for the entry. And then in the output, we have the relationship between the human interactome and the virus interactome. That's something that was recently obtained by the group of Krogan. Or we can have other kind of pathologies, such as the acute respiratory distress, which is an outcome that gives you the information on how it is affecting the virus. So in the next slide, I will show the results that we have obtained. So first, with the use of TPMS, we have been using all potential pairs of drugs that are underused commercially and are stored in drug bank. And then as the output, we use the virus host network. So we are trying to obtain the highest impact on this network. And then we rank all these pairs of drugs, and we focus on those drugs that were involving affecting furin, is one of the targets in the entry. This happens to be pirfenidone, and then the highest score is gotten for the combination with melatonin. Then on the right side, we have what we did with Gildify. So now we know the input, so we put both targets as an input, and we obtain the connection of the whole networks of both drugs, from where we can get what are the relevant pathways or the regimen of functions. And also using TPMS, we can see what are the mechanisms of action more relevant for the connection with acute respiratory distress. So among them, we can obtain using Gildify, and then this study with TPMS. Some relevant properties that comes out from this study are the characteristics of these two drugs that are anti-inflammatory, antioxidant, antifibrotic. So we think that this can be used just to relieve the severe pulmonary complications coming from COVID. And with this, I'm done with the presentation, and later for the questions, we will also have Mireia Coma from Anaxomics that can help on the answers. Thank you.
Okay, much. Although I hope you is listening me well because I have troubles with microphones. Now it's done from just and doesn't done. How you viruses BioRegion?
Hello, good afternoon. Can you hear me well?
Yeah.
Okay, so to achieve the 4 objectives of our project that has been funded by the degrees, we have followed 2 main strategies. One has been to sequencing the whole genome that now is working properly well in the laboratory of Dr. Andres Anton. And also we have followed a deep sequencing analysis of some genes of the SARS-CoV-2. Next slide, please. And specifically, we have focused our attention in the sequencing of the spike gene that codifies for the spike protein. The spike protein is composed by 2 regions. The region 1, the S1, includes the receptor binding domain, and the S2 is the one that fixes the spike to the surface of the virus. To sequence the whole spike, we have designed 13 primer pairs. Each primer pairs, we call it an amplicon. So next step, next please. And we have studied the spike of 18 patients. And the result that we obtain is something like this graphic that we make a zoom like this. In this graphic, we show, we have studied the deletions. Each column, the blue column, represents a deletion. The width is the number of nucleotides that are lost, that are deleted. And the height is the number of reads of sequences that we obtain. In the right side of the graphic, we represent the percentage. Next, we have studied more than 200,000 complete spikes per patient. And the results that we have obtained are represented in these boxes, in the 13 boxes that represents the 13 amplicons. Inside of each box, we represent the deletions in each of the patients. And I want you to highlight the amplicon number 7. Here, as you can see, there is an accumulation of deletions in most of the patients. And all these deletions are observed exactly at the same positions, which is upstream, but very, very close to the S1 and S2 cleavage site. Next, please. So this has led us to propose a hypothesis. In the left side of this graphic, you can see the normal viral cycle of the virus that finishes with the production of a genome of an infectious virus that is secreted by exocytosis. And in the right side, you can see that when there is a deletion, these deletions causes the appearance of a stop codon, and then this generates the translation, the production of a truncated spike protein that is composed by the subunit S1. And as this S1 is the external part of the spike protein, it is probably that this peptide is soluble, and we propose that this can be secreted and it can do the best that it can do. So it's binding to the ACE2 receptor. Next, please. So this work, next slide, please. This work has been recently published in Emerging Microbes and Infection. It's the work of different groups on BioRegion, of Campus BioRegion. And the highlights and implications of our results is that natural deletions are a source of viral variation, that there are some hotspot deletions upstream and very close to the S1 and S2 cleavage site, that these generate truncated S proteins. And we suggest that these proteins, this is a kind of a strategy that we have called the don't burn down the house, in which the free S1 protein may compete with the viral particles for the ACE2 receptor, and thereby attenuating the tissue damage, and this helps the virus to produce viruses and be transmitted more easily. In addition, it is possible, or a possible consequence is that this free S1 protein may bind to host neutralizing antibodies, and this has helped us to start a new line of research trying to find these free proteins in infected patients. And thank you very much for your attention.
Thanks very much, Josep. And now is the turn from Joaquim Segalés, please.
Thank you very much, Anna, for the presentation. Good afternoon to everybody. Well, there's always an animal side of everything. So from this point of view, this part, it will include the part which is much more related with the One Health concept, because at the very end, at the start of these pandemics, it was a lot of unknowns, and some of the unknowns were related with the potential zoonotic origin, but another one was which kind of animals were susceptible to SARS-CoV-2. At the very end, it was very important to know if there are several potential species which are susceptible, and especially those that are able to recapitulate the problems that the human beings are suffering from COVID-19. So by February already, March, the World Health Organization established a COVID-19 animal model working group. And very recently, this working group has published a paper, which you can see on your right-hand side, regarding animal models for COVID-19. So in our case, instead of presenting rather than a project, it's much more a platform related with the animal model development, specifically oriented to work with SARS-CoV-2 and COVID-19. Next slide, please. In fact, the major objectives of this platform was to develop such different animal models with the specific objective to study pathogenesis and immunopathogenesis against the virus, and very importantly nowadays, and especially within this section of repurposing drugs, on the application of such animal models to test antiviral drugs, but also antibodies, and very importantly, vaccines. In fact, the most, let's say, precise objectives would include the development of animal models able to recapitulate what's going on in the human beings. This is the difficult part, taking into account that the human beings, they have a very broad spectrum of clinical signs as well as lesions, and also to try to find animal models usable for immunogenicity studies when developing vaccines. Next slide. So far, and since March, we have been working on the biosafety level 3 laboratory and animal facilities at IRTA-CRESA. Those are very special kind of facilities that allows us working with SARS-CoV-2 and do infections, not just in vitro, but also in vivo with this kind of virus. In fact, we have been working for more than 6 years already with MERS coronavirus, another important zoonotic deadly coronavirus. Well, so far and since then, we have established the hamster model with the golden Syrian hamster. This is a kind of animal which is naturally susceptible to SARS-CoV-2. In fact, the model is relatively simple because it consists on the international installation of a certain amount of virus which could vary between 10 to the 4 to 10 to the 6 TCID50 per animal. And the readout of the system is excellent because it includes 3 major legs, which is the clinical one because of losses weight, the pathological one because they develop moderate levels of pneumonia, interstitial pneumonia, and finally from a virological side because the viral load, especially in the respiratory tract, namely upper respiratory tract, nasal turbinates, and lungs, is a very important readouts. Moreover, we have been able to establish a kind of system of infection and reinfection with the hamster model, and we observed that certainly the animals can protect against the lower respiratory tract open reinfection, but not the upper respiratory tract. We also set already the humanized AC2 mice model with a dose of 10 to the 3, 10 to the 4 TCID50 per animal, again for international installation. And we also recapitulate several different issues which could be certainly comparable only to a certain degree with humans. There's a loss of weight, so there's a clinical outcome. However, there are very significant neurological signs, which so far it's not the typical presentation in humans, as well as pneumonia and huge viral load in respiratory tract as well as brain. And finally, we set a swine model we realized already that pigs are not susceptible to SARS-CoV-2 infection. However, when we did experimental infections by means of parenteral inoculation, namely intravenously and intramuscularly, those animals were able to seroconvert. So pig probably can recapitulate to a certain degree a potential immunogenicity model, and take into account that pig could be a physiological and immunological good model for human, so we have to take into account that could be of interest. Next slide, please. So which is the research impact that we expect from those animal models? Of course, the possibility of do reinfection studies, or even to do multiple passages, or even the possibility of studying aerosol transmission, which is quite hotspot nowadays. Of course, we can model SARS-CoV-2 transmission in different settings, and at the very end is the possibility of recapitulating as much as possible the COVID-19 forms. So far, we have been trying, especially with the hamster model and the mice model, young animals. What could happen if we are using older animals or animals suffering from comorbidities, for example? And very importantly, I would say, and this is the short-term most important issues at present, is the use of those models as preclinical study technology. Because at the very end, if we have to test vaccines, antibodies, or antiviral drugs, it will be very, very important to use such models in all cases. And with that, I would like to finish just to say that this kind of platform has been obtained at the very end with the collaboration with Directorate together with Barcelona Supercomputing Center and IRSI Caixa in a kind of big consortium that has been also participated by Grifols. So with that, I thank you very much for your attention. Thank you.
Thanks, Joaquim.
Thank you. Hello, everyone. And today I will be presenting the project of a dual strategy for inhibiting the infectious cycle of SARS-CoV-2, which is supervised by Jana Selent and in collaboration with Thomas Espetifnis and Maria Vadhua from Interax. As the name suggests, our project takes a dual approach for targeting and inhibiting the entrance of the virus into the cell using already approved drugs and candidates in advanced investigational stages. As you know, SARS-CoV-2 viruses uses its surface protein called the spike protein to bind to the human receptor ACE2. Subsequently, the human serine protease TMPRSS2 facilitates the virus entrance into the cell. So, our first strategy consists in blocking a specific region of the spike protein, called the receptor binding domain, that is used to attach to the host cell. In order to find compounds that could inhibit this attachment, we performed a virtual screening consisting in protein ligand docking into the interface between both proteins, the spike proteins and the cell ACE2. Our second strategy consists in inhibiting the serine protease TMPRSS2, which catalyzes the entrance of the virus into the cell. For this target, we could not use the docking approach given that it does not exist a crystallized structure for this protease. So we decided to perform a virtual screening using a ligand-based pharmacophore approach based on ligands that are known to inhibit this protease. This pharmacophore approach consists in detecting common structural features that are related to the protease inhibition and that can be used in a next step to screen for novel compounds that match also these patterns. In the remaining slides, I will explain the main steps of the project. The first step was to perform a virtual screening with the methods already explained, the docking and the pharmacophore-based approach. We screen against a database consisting of the merge of DrugBank, PubChem, and also other metabolites found in SIG15 database. The second step, after we found some candidates, was to perform molecular dynamics binding simulations of the top drugs against their targets. Then, the third step was to look for antiviral activity against SARS-CoV-2 in the bibliography. In this step, we found that 3 of our top hits were antivirally active at safe concentrations, as Riva et al. Stated in the recently published Nature paper. The candidates that emerged from our screen are highlighted here in red. Apelimod and astemizole performed excellently in our docking results, which we can see in the next figure, scoring higher than 97% of the molecules in the docking. In addition, we found flumatinib with an even higher score than that. Finally, in our fourth step, our colleagues of Interax, in the next slide, have established an assay to measure viral infection using SARS-CoV-2 pseudovirus. This pseudovirus carry luciferase RNA that, when the infected cell, is expressed inside them. This way, we can quantify how many viruses enter the cells, and this way test whether our top compounds inhibit the enzyme's mechanism or not. So using this assay, we obtained our first evidence that apilimod inhibits the entry step of the infection. In the following, you see a quantification of virus entry measured as luciferase activity showing on the y-axis. The more luminescence means the more virus have entered the cell. On the left, you see a control experiment with an ACE2 antibody. The higher the antibody concentration, the less virus enters the cell. Meanwhile, on the right, you see our results for apilimod, which reduces virus entry by approximately 80%, which we think is very promising. So currently, we are now in this phase of confirming these results and also testing other several promising candidates. That's it for now. Thanks you for listening.
Okay, so thank you very much. We had planned that Ana Meseguer will host a short debate, but I'm afraid that we have sound problems. So we'll just thank you, Maria, thank you, Baldo, Josep, Kim, and Alejandro for your very interesting presentations. I think that we should, now it's time for the audience to vote for the beautiful science and for the game changer project among these 5 projects that we have seen now. So if you can, the guys who are managing the Zoom in Girona can really put the screen with the voting and close the presentation and plug in the screen with the poll. Also, I would like to remember the people, the audience, that there is a chat screen where you can also send questions to the panelists. It's not just us who are going to ask the questions or open a little bit of a debate, but you, the audience, very much invited to pose your questions to the panelists. So let me start with Maria Jose. You've mentioned that anti-ACI-AC2 antibody does not block viral entry in all patients equally. Does it mean that there are other receptors playing a role in viral entry? What's your take on that?
So I don't know the answer yet, but we are working on that because it was a very interesting finding. It might be that there are some other receptors that are mediating viral entry, or maybe some other isoforms of the ACE2 receptors that are expressed in different cells. So I don't know the answer, but we are working on that.
Okay, thank you. Of course, it's one of the key questions that everybody is wondering about these days. Next, Baldo, do you think, or is it possible that melatonin alone might have an effect on the evolution of the infection, or it has to be a combination, and why a combination?
We presented this as a combination, but actually this is an interesting question that it was posed by an email by an Indian clinician. And I know melatonin. It's appeared also in the work of Krogan as one possible drug to be used. And I think Mireia, I don't know if she can be here too, but in Anaxomics they also studied more deeply on the use of each one of the individual drugs. Perhaps she can comment on that if she is in, but I'm not sure.
No, I don't think we can have Mireia here.
Okay,
so it's so complicated. You cannot imagine how complicated it is on the backstage to organize this meeting. I'm sorry.
So.
I think we are doing we're doing quite well, but it's difficult to plug in people from so many different places. So thank you. I mean, please go ahead, Baldo. Go ahead.
Yeah. No, I know they did study each individual case, so that's why I know they have more deeply analyzed each one of them separately. But the work was done on the combination. It appeared melatonin and pirfenidone, but there were also many other pairs that came out of this work.
Okay, excellent. So next question. I mean, I have, of course, one question for each one of us. And please, the audience can also pose your questions. Or, of course, this is one of the goals of this webinar, is that everybody gets to know everybody. Maybe you have never worked together. You don't even knew that Josep existed from Baldo's perspective. I don't know, maybe yes, maybe not, but it's important that to have this sense of community. We are all together from very different places, universities and hospitals. We are all together today here, trying to solve a common nightmare for all of us. So it's also good to get to know each other. So please, the panelists can pose also questions to the other panelists. Feel free to do that. Josep, you've mentioned the strategy that don't burn down the house is something that the virus has acquired during the pandemics. I mean, what do you mean by that?
Yeah, well, the answer is that we don't think that the virus has acquired it during the pandemics, because the samples that we have studied came from the beginning of the pandemics. So they were taken in March 2020, so when it started here. So that we think is that the virus was carrying this strategy from the beginning. And is it possible that this kind of strategy can be selected in different, during the evolution of the virus, jumping from animals to humans or whatever? Because it's something that can be quite general, not specific of the humans in this case.
Okay, thank you. Yes. Joaquim, Kim, I mean, do you think that other animal models can be of interest? I mean, besides rodents? I don't know.
Yeah.
Monkeys, mini pigs. I mean, what other ideas, or what's out there in this field?
Well, at the very end, a number of animal species have been tested for susceptibility to SARS-CoV-2. However, just a minimal number are naturally susceptible. And among them, for example, besides the golden Syrian hamsters, we have the ferret, as well as the mink. So those are animals that they can recapitulate to a certain degree a certain disease. However, it's extremely mild and sometimes no clinical signs. Like the cat, the domestic cat and other felids are susceptible to the infection. However, they do not display almost clinical signs or minimal ones. The other ones, which of course are quite far away from our possibilities, or for most of us, is the non-human primates. Non-human primates, at least some of them, recapitulate rather nicely as well the disease in humans, a kind of moderate disease. However, if the specific question would be, can we get an animal species recapitulating a really severe form of COVID-19? Nowadays, we do not have it. This is something that might be in the future will come, but might be we will need also animal species suffering from, might be comorbidities, co-infections, or even elder animals, which is not easy to work with.
Okay, thank you. Thank you very much. Very, very interesting, your perspective on that. I will pose one last question to Alejandro, and we have one question from the audience, and we will move to the next block, because I mean it's going to be an intense afternoon. We have many presentations. So this question is for you, Alejandro, or somebody from your team that can really chip in. You've mentioned two strategies to inhibit the infection of SARS-CoV-2, blocking the spike protein or inhibiting the serine protease. Which is your preferred strategy? What are the advantages or disadvantages of each one of them?
I think that maybe the first strategy, as we are focusing on virus protein, has less side effects because we are not inhibiting natural processes in human cells. So in that way, I think that the first strategy will have less side effects, so will be more useful. And also, as it is a structure-based approach, it is specific for the spike protein. Probably will have more direct effects than doing a ligand-based approach that is maybe more general for a wide range of targets. So strategy one, I think, would be the most promising one.
Okay, thank you very much.
Thank you.
I think if it's possible, can we insert the poll, the voting screen, so everybody can start voting very quickly? The preferred beautiful science and game changer project. Well, whilst we wait for that, there is one last question that would like to pose. I think it's for Maria. I mean, there are many questions from the audience. We don't have time to ask all of them to the panelists, so we'll just go for one of the many of the several questions. Is as follows. I mean, is the idea of finding a drug? This is from Giulio Rosati. Is the idea of finding a drug blocking ACE2 receptor involving people solved? I mean, are there drugs in the clinical trials blocking ACE2 receptor under study, or is this is just an idea that we have discarded for the time being?
That's a question for me?
Yes.
No. Oh, yeah. To my knowledge, there is not. But I mean, the drugs that has been described in the literature, they are trying to block or the S protein or the interface between the S protein or the AC receptor. But I'm not 100% sure of the answer.
Okay. So I don't know if the audience is seeing the voting poll. Okay, so people can vote. Sorry, it's because I'm seeing a different screen. So I understand that people can vote now. So please remember to vote in two categories. I mean, the beautiful science. Which of these 5 projects did you like the most? Do you think were more innovative or creative? And which one of the projects is the one with more chances of being a game changer in the short term and really change our lives and the global course of the pandemic? Remember to scroll down the screen, because the voting, the screen is long with the different projects, so the voting button is at the end. Excellent. So I think everybody has been able to vote. Beautiful science, game changer. So thank you the panelists. Thank you, Maria, Baldo, Josep, Kim, Alejandro, for your beautiful and exciting presentations. I hope you're the winners. Please stay with us until the end. Also, please vote. Again, there is 2 votes ongoing at the same time, so we like to make things as complicated as possible. We enjoy complex things. So there is 2 votes, beautiful science, game changer, and at the same time, in parallel, we will be voting or betting, more than voting, betting, which is going to be the first vaccine to reach approval by the European Medicines Agency. Oxford team, I think, is on top. Moderna is following closely. Couple of Chinese projects behind. BioNTech, Novavax, or you can even vote others if you think that those are not those are not the front runners. Okay, so thank you very much for these panelists. Let's move now. Thank you all.
See you.
Thank you.
Let's
Bye.
move now to the second block. I mean, 1 down, 3 to go. The second block for the second section is about new therapies. This first one was about repurposing. The second one is about new therapies. And the chair of this second section is Ignacio López Verdaguer, Director of Science and Research from La Caixa Foundation.
Dr. Juana Diaz from Pompeu with RNA-based therapy therapeutical platform. So thank you very much to everybody, and we wanted to be brief because we would like you, the audience, to make as many questions as possible. So without anything else, I would like to start with Laia, and I'm sorry I call you Laia because we know each other quite well. So please, the floor is yours. Well, this is not a mistake. It is just because so you can remember all the slides, so you can vote properly later.
Okay. Okay. So thank you for the organizing these talks. As you know, I am leading this project together with Professor Antonio Torres from the Hospital Clinic. We are settled in IDIVAPS here in Barcelona, and we have a big challenge here. In which we are identifying SARS-CoV-2 epitopes in order to be able to produce monoclonal antibodies for treatment. We have used overlapping predictions of 3 different methods in order to find those epitopes with higher accessibility for the antibodies. In parallel, we have collected as well serum samples from post-COVID asymptomatic, mild, and severe patients. And we are, by a Luminex assay and immunoassay, we are identifying which are the epitopes that have the highest binding to the antibodies that in the reality our population with COVID have produced. With this, we will isolate these antibodies through high-performance liquid chromatography, and we also will describe the X-ray quaternary antibody structure and the sequencing by tandem spectrometry. We can move to the next one. Okay, as you know, in spite of the high similarity with SARS-CoV, the SARS-CoV-2 has completely created a pandemic, and because of the high transmissibility and infectivity. In the top right of the slide, I put some examples of these epitopes that we have generated, so you can see how are they located in the spike protein, and see that 7 of them, of these examples, are allocated in the region binding domain, which is the domain that reacts with the receptor. Going into a deeper detail, what we have done is we have created we have coupled our peptides with a polymer, PEC12 and biotin complex, and we have coupled this with magnetic beads plus avidin in order to perform our Luminex-based immunoassay. And this assay is what is telling us which are the epitopes that are producing highest binding to the antibodies of our post-COVID population. See below, for instance, how the peptide epitope 2 has produced much higher luminescence in much higher number of patients compared to the epitope 3. And this is what we are doing. We are identifying potentially 5 or 6 best epitopes candidates in order to produce these monoclonal antibodies. We can move to the next slide. And on this furious race against COVID infections, I would like to highlight that, of course, we are working in a completely accelerated timeline. You know that in a typical timeline for a vaccine is 16 years or 15 years, and the goal of the entire scientific community is to have available new treatments and new vaccines in short during the 2001 or early 2002. So this is a completely difficult challenge. But what we have up to now is what we have learned from, for instance, from current therapies, and regarding antibodies, we have the use of convalescent plasma. In the United States, more than 20,000 patients have been treated with convalescent plasma of post-COVID patients successfully with very few side effects. And we know now that it's very important that these antibodies, of course, are against the target, that we administer enough amount of antibody, and that this administration has to be early, optimally in the first 3 days of hospital admission. So these are good news for us, and we hope that this cocktail of monoclonal antibodies can have prophylactic and also therapeutic benefits for instance, healthcare professionals or high-risk and severe patients. There are currently 2 phase 3 trials that I would like to highlight that you probably know. One with a combination of 2 monoclonal antibodies, and then we have another one with a single monoclonal antibody of Lilly. And just to finish, we need to bear in mind that these monoclonal antibodies have to be prepared to avoid the mutational escape that is obvious that is occurring in this coronavirus. Let me mention that, of course, we are a big consortia. We are collaborating with IS Global, with the SIG, with Barcelona Supercomputing Center, and with IRB, and I am very happy to have all these people in the team. Thank you very much.
Well, thank you very much, Laia, for presenting this very interesting project. And now we go to Giuliana Magri from IMIM. Thank you very much. Dr. Magri, you are muted now. Okay.
Yeah. Okay. So good afternoon to everyone. So today I'm going to briefly introduce our project that has been recently granted by the Department of Health of Generalitat de Catalunya. So the work team for this project included 2 very talented researchers at the B-Cell Biology Lab, Dr. Leire de Campos and Dr. Sonia Tejedor, a clinician at Parc Salut Mar, Dr. Judith Villar, and 2 data scientists at GRIP, Dr. Laura Furlong and Dr. Janet Pinheiro. So the idea behind this project is to use the patient's own immune response, humoral response, as a template to develop and engineer in vitro neutralizing antibody to SARS-CoV-2 that can be used as a therapeutic drugs to block and prevent the infection. So the first goal of this project is then the characterization of SARS-CoV-2 specific B cells and antibody dynamics during natural infection in moderate and severe COVID-19 patients, as well as in convalescent individuals. And then we will use the information generated from this initial analysis to establish the best pipeline to generate in vitro safe and effective human recombinant virus-specific neutralizing monoclonal antibody from the PBNC of convalescent individuals. Next slide, please. So we initially perform, analyze and profile SARS-CoV-2 specific antibody response in a serum sample from a cohort of patients with moderate and severe COVID-19 disease. And we do that by performing isotyping and in-house developed isotyping and subtyping ELISA using the spike, the receptor binding domain of the spike protein, as well as the nucleocapsid protein as a substrate. So as it's shown in this heat map, based on the SARS-CoV-2 specific antibody profile, we were actually able to cluster patients into different groups Then we apply a machine learning method to define the minimal set of immune features that distinguish patients with moderate from severe COVID-19 disease. And we found that indeed patients with severe disease have a significantly higher RBD-specific IgG1 and IgG3 antibody, whereas patients whereas a dominant RBD-specific IgG2 response associated with a more favorable clinical outcome. Next, please. So in parallel, we are also analyzing in peripheral blood lymphocytes by conventional and spectral flow cytometry, peripheral lymphocytes of patients in the acute phase of infection, as well as 2 months from symptoms onset. And the preliminary results from this initial analysis suggest that indeed acute COVID-19 infection is associated with transitory changes, mostly with transitory changes in circulating lymphocytes, including expansion of activated CD4 and CD8 T cells, as well as a huge expansion of plasmablast. Also, we observe an expansion of extrafollicular memory switch B cells that tend to persist also during the convalescence phase of infection. And this can be relevant for the development of neutralizing antibody. Please, next slide. So to develop human recombinant SARS-CoV-2 specific neutralizing monoclonal antibody, we have set up a protocol to detect RBD specific B cells in the blood of convalescent individuals. So as you know, the receptor binding domain is the part of the spike protein that is responsible of the binding to the ACE2 receptor on target cells. So often antibodies that recognize the receptor binding domain are actually able to block the entry of the virus and are neutralizing. So as you can see in this slide, we can able to detect RBD specific B cells in convalescent individual. Those cells actually persist up to 6 months after infection. And also we found that mainly consist of memory IgG positive B cells. Next, please. So we use the same cohort to develop a first batch of virus-specific monoclonal antibody. So we stain PBNC from this convalescent individual with a mix of antibodies that are specific for B cell marker, as well as with our fluorescence labeled RBD probe. Then we single cell sorted RBD specific B cells into 96 well plate. Then we got the cDNA from each one of these cells, and by PCR we have amplified the variable region of the immunoglobulin heavy and light chain. So this is unique for each one of the cells and is the part of the antibody that is responsible of the binding to the antigen. So then we are sequencing right now these amplicons, and then with the selected amplicon will be vector ligated into IgG encoding plasmid, and then co-transfected into 293 mammalian cell line. So each one of these cell culture will produce in their supernatant a monoclonal antibody that can be tested for reactivity and also for neutralization activity by in vitro and in vivo assay. So next, please. So we think that our initial characterization actually will lead to the identification of the antibody property that best correlate with disease recovery and protection, as opposed to disease progression, including the induction of potential induction of inflammation or antibody-dependent enhancement of infection. And this will guide us the definition of a very straightforward strategy for the development of virus-specific monoclonal antibody, including the selection of the best high responder, the selection of the best virus-specific B cells, and when we have a candidate, also the antibody optimization to maximize immune protection and safety. Please, next. So right now, the generation of a virus-specific monoclonal antibody is indeed recognized as one of the most promising approach to fight COVID-19, and we think that may constitute an important therapeutic bridge until a vaccine becomes available. Also, once a vaccine is available, neutralizing antibody can still be useful as may provide protection in immune-compromised individuals. And despite the high cost, if we are able to generate a neutralizing antibody with outstanding neutralization activity that can be used at lower dose, and this will reduce the economic burden on the health system. So, thank you very much.
Thank you very much for a very clear presentation. And let's go to Dr. Rafael Mañez from Belviche, please.
Well, good afternoon, and thank you for the invitation. Well, we'll be presenting certainly a new approach, totally different approach to generating protective immunity against SARS-CoV-2, that is basically the removal of non-neutralizing antibodies. Next slide, please. We know that antibodies are one of the main mediators of the immune response against all the infections, and certainly the SARS-CoV-2 is not really the exception. Once we have the infection, we generate different antibodies against the virus. As we are showing here, we are producing antibodies against, for instance, the spike protein, that is the one that interacts with ACE2 receptor to infect the new, affect the cells. However, there is a well-known phenomenon that, particularly in the case of viruses, when the level of antibodies are low and not all the receptors that cover, may happen this phenomenon known as antibody-dependent enhancement of infection, in which what occurs is basically that the antibodies really, they cannot neutralize the virus, and what occurs is that the antibodies, there is a reactivity between the Fc fragment of the antibodies with the Fc receptors in macrophage and monocytes, and really facilitate the infectivity. This has been very found in all the viruses, particularly in all the coronavirus infection, and also in all some flavivirus, like particularly dengue disease. Next slide, please. Along with the specific antibodies, we are also antibodies known as natural antibodies, are polyreactive antibodies that also play a role in the identification and binding to viruses. And some of these antibodies may also be really act lack of neutralizing capacity, and may perhaps act as also facilitator of infection. There have been already several studies, the most recent one, this one published in the New England Journal of Medicine last week, showing that there is a clear relationship between the blood group type and the development of severe diseases in the coronavirus infection, and particularly that blood group A, in other words, people having anti-blood group B antibodies, really have a higher incidence of severe disease. Next figure, please. Our group had been working for several years already in a strategy to remove different antibodies that are really harmful antibodies or deleterious antibodies to the individuals. We have developed a platform constituted by polymers to which we can attach different antigens, the specific antigen of the antibody that we want to remove, and that so far are mainly sugars. And these glycopolymers can be injected intravenously or subcutaneously. They initially remove the antibodies, that circulated antibodies. The immune complexes that we generate are removed, metabolized, in the liver, are removed to the kidney. And also, they also bind in the lymph node and spleen new the B cells producing these antibodies. One means that we only are not removing circulated antibodies, we are blocking the production of new antibodies, in this case, those specific antibodies that we are interested. Next slide, please. Our approach that we've been working is we are trying to identify those non-neutralizing antibodies binding the different structure of the virus, and particularly those anti-carbohydrate antibodies that bind the shield that all these viruses, and particularly coronavirus, the glycan shield that this coronavirus that they have. We are using the glycan array technology that allows for identifications. We have a panel with 750 different carbohydrate sugars, and this allows us, using inhibitory assays, to identify those antibodies that bind the different viral structures and which one they lack of neutralizing activity. Once we have identified these antibodies, we can use our polymer platform to generate the glycopolymers. And then next figure, we can, what we are using is try to see whether the specific removal of some of these antibodies, as one suggested so far, the anti-blood group B antibodies, could really enhance the neutralization capacity of human serum against the SARS-CoV-2 virus. The next slide. We think that summarize is that, well, we are proposing really a game changer, a way to treat infections. It's probably a change of paradigm. It will be the first time that we are proposing to generate protective immunity against infection by removing existing antibodies in a state of producing no ones. Our treatment could be both preventive and therapeutic, because certainly we can remove those antibodies generated during the infection that may also act as non-neutralized antibodies, and of course, those natural antibodies that really act and facilitate the infectivity. And one of the clear advantages, we can remove, these are all the antibodies in 1 hour after the injection, what means then we are able to generate an immediate immunity against the SARS-CoV-2. And this is certainly one of the advantages and why we can be used, our asset can be used either as preventive or therapeutic treatment. It's safe. We have non-human primates treated for prolonged periods with the removal of specific antibodies with no side effect. And we already have well-defined dosage and also way of administration that, as I said before, could be either the subcutaneous or intravenous administration. Thank you very much for your attention.
Well, thank you very much, Dr. Mañez. So let's go to the next project. That would be Dr. Núria Montserrat from IBEC.
Okay, good afternoon to everybody, and thanks for the invitation to present part of our work. I hope to convince you about the utility of bioengineering and how to grow tissues in the laboratory to start to understand and target SARS coronavirus 2 infection. Something that we think it's wonderful, next please, from the nature of pluripotent stem cells is that these cells that can be found naturally in human embryos, and we can also derive this kind of cells in the laboratory, is that these have the capability to give rise to all the cell types in our body because of their inherent properties. This knowledge has been very useful in the laboratory to develop in the next animation organoids. These organoids are three-dimensional like tissue structures that are composed of multiple cell types. And the magic of these systems is that the cells can be encountered in a very similar fashion as we discover the cells in the organ that we like to mimic in the Petri dish. Importantly, different laboratories, including our laboratory, we have been also showing that the cells that are in these structures are also organized in a spatial fashion very similar to the one that we find in the organ. And in the next animation come the major questions. Can we profit from these mini organ-like tissue cultures to start to understand how SARS coronavirus 2 infects human cells? And also, can we think in exploiting this technology to further understand how to model COVID-19 disease? So these questions were important for us already in February when it was discovered, especially in China and different countries all over the world, on the sequence, genomic sequence of SARS coronavirus 2 and the similarity of SARS coronavirus 2 with SARS coronavirus 1 that we know and we knew already that uses mainly ACE2 receptor for entering into the cells. So our hypothesis was that we could block the entrance of the virus by using a compound that was already tested in a first one clinical trial in healthy population. This compound is a soluble recombinant protein for ACE2, mimicking specifically the chemistry of this molecule. So then we hypothesized to use these microorgans, kidney organoids, and also vascular organoids to infect the organoids with our collaborators in Karolinska Institutet in Sweden, and to check for the possibility to block viral entrance and replication. And this is what we show very early in April, that we could infect these cells, majorly because human organoids contain proximal tubular cells, very similar cells of the ones that we find in the kidney. And once we infect the organoids, we could treat these organoids with this clinical tested compound that nowadays is in a second phase clinical trial in 6 different countries. So if you put the next animation, it's more to tell you that we did publish this collaboration very early in April, beginning of May, and now just very recently, we published on the efficacy of the compound in patients in the journal The Lancet Respiratory Medicine. And nowadays, what we are doing with CRISPR-Cas9 is to delete or completely abrogate the expression of other putative receptors for SARS coronavirus 2. Not only ACE2 has been involved in the capture of the virus, also other molecules, as CD147, for example, and other lectins. So with CRISPR-Cas9, we are having the possibility to totally delete or abrogate the expression of these molecules. And in the video that is there just in the next animation is for showing to you how an organoid looks like infected by the virus in yellow. And we are now testing on the effect of different compounds in the background of engineered organoids, also using genetic tools. In the final slide, in the third slide, I would just like to highlight on the possibilities, the theoretical possibilities of using kidney organoids. We can start to study SARS coronavirus 2 host interaction, for example. We can explore for systemic conditions. We are also having other projects in collaboration with our partners at Karolinska Institutet, also in the University of Heidelberg and in Vienna, in where we are testing on other comorbidity-related issues like diabetes or hyperlipidemia, on how it impacts on the infection per se on these kidney organoids. Using bioengineering, we have been really taking advantage of different approaches for growing the organoids, and in the last animation is just for showing to you that we can exploit kidney organoids together with CRISPR-Cas9. So I think we have, okay. So in the last part of the slide, you can go all the way, because otherwise it's very difficult for me to speak. Okay, thank you very much. So it's more for telling you on not only on the scientific impact of this kind of application for studying COVID-19 disease, but also on the social relevance of this type of project. It's clear that from the scientific side, we are working in an emerging field. We are combining different tools that are innovative with regards of the use of organoids and CRISPR-Cas9 engineering. But for us, what is super important is on the social relevance of the project. So we are working together with different institutions. We are working always keeping in mind the European community, research community indications in terms of gender balance, including in our research and also in our working teams. And for the industrial side, on the industrial impact, all of the different issues that we are playing with in the project have also a long-run framework. So I would like to conclude with saying a big thanks to you and hoping that I convince you on the possibility to make use of mini organs to study SARS coronavirus 2 infection. Thank you very much.
Well, thank you. Thank you, Núria Montserrat. So, and last but not least, Juana Diaz. That would be the last presentation. Dr. Juana Diaz from Pompeu Fabra. Here we have the presentation, so the floor is yours. I don't see you in my screen now.
Can you see me? Can you hear me?
Yes, perfectly.
Okay, great.
Go ahead, please.
Hi to everyone. I would like also to thank the organizer for having this nice meeting. It's great to have a global view of what is going on in Barcelona related to COVID-19. So in my laboratory, we have developed a novel RNA-based therapeutical platform that will allow for ultra-fast responses against viral pandemic outbreaks. These fast responses are super important because for outbreaks not to become pandemics and for pandemic ones to be able to control them. Next slides, please. Our platform is based in 4 components. First, an artificial intelligence-based software that we design in-house that allows us for a fast design of molecules that inhibits the virus of interest. This, as an example of this accelerated development, here down in the, I don't know if you can see my mouse, I guess no. So in the down part, in the accelerated development, you will see that in day 1, after we know the sequence of SARS-CoV-2 is published, then it takes 7 days. It took us 7 days to design 20 candidates, 20 RNA-based molecules candidates. Then at day 14, we have already these molecules produced. And finally, at day 18, we have 3 candidates that were validated with a highly inhibiting capacity. And this percentage of success is what we have been finding for different viruses. So another characteristic, another part of our platform is that the RNA-based therapy that we design, in contrast to others, they are highly stable. And this is now covered by a European patent. Third, we have developed a method that it has a very low cost production of these molecules, as low as €2 per microgram. And finally, we can deliver the molecules in a very efficient way by nanoparticle system that is already approved to be used in humans. So therefore, as a competitive advantage of our platform, I would like to emphasize the high versatility, because we can apply it for novel outbreaks, that it has a low development cost, that is, the molecules are highly stable, and this will assure a reasonable administration time between doses in patients. And there are also two main features I would like to, that we find very attractive, is one that it is designed in a way that it hampers the emergence of escaping mutants, and this is the typical problem against antivirals for most of the viruses, the RNA viruses. And last, that we could design RNA molecules that within one molecule may have or can have broad spectrum activity that could be across serotypes or even among different viruses. May I have the next slide, please? So as an example of the in vitro efficacy, here you will find a graphic where we have the percentage of infectivity related to a negative control and to the 3 molecules, to the 3 hits that I told you about. As you can see, the activity was almost completely gone after treating with our design molecules. This was done in vitro. Then, related to stability, in vitro assays, it shows us that the stability of the molecules is between 48 and 72 hours. That does not change the stability, so we can, every 3 days, we'll have to give new molecules to the cell. And in vivo, in mice models, using these nanoparticle delivery methods, we have seen in different organs that is stable by 48 hours. And this is now seen as well recently, today, in lungs, and even in lung is even more effective. May I have the next slide, please? So to end, related to the impact, I would like to emphasize that the use of the antiviral therapy will have the greatest clinical impact before the illness progresses into the hyperinflammatory state, that is the typical symptom of the later stages of the disease. And that reducing the viral load within 2 days will radically shorten the contagious period. And in line with this, the objective will be to get an isolation of the COVID-19 cases pharmaceutically rather than physically. This will have a highly economical impact, and also it will disrupt the chains of transmission. Finally, I would like to emphasize again that this platform is very versatile and could be rapidly applied to other novel viral outbreaks. And with this, I thank all of you for your attention.
Thank you very much, Dr. Diez. So thank you very much to all the panelists. I am sure that we will find amazing science and game changers in the other panels, but my bet would be that we can find both of them here. Thank you very much for the clarity, but as well for the very innovative approach that you are using in your different projects. I am being said that, well, of course, first of all, that these votings, you can do them now during the questions and answers of this panel. As you heard before, Jordi is very much interested in this polling, so please go ahead with the voting. But as well, now is the moment for questions and answers, so if you have any, please do them in the chat. In the meanwhile, I see that here in the panelist chat, there is one question from one panelist to another, and maybe we can share this one. Dr. Diaz was asking something to Nuria Montserrat in relation to the organoids. Maybe you can share that with the rest of the audience.
Oh yeah, I would like to ask you, related to the organoids, it was not clear for me if these organoids mimic some kind of organ specifically. I mean, they could mimic lung, they could mimic, or it's just general. Could you please explain to us?
Yes, the technology of organoids, it's been for the last 5 years, I think, one of the major booms in the field of stem cell or cell biology. We can derive organoids from adult stem cells or from pluripotent stem cells. In the laboratory, we are focused on developing organoids from human pluripotent stem cells, the ones that can be derived into any cell type of our body. But other groups in the world are also deriving lung organoids, and this is not new. The work has been advancing quite a lot using mouse embryonic stem cells, and you can also find work on lung organoids from human pluripotent stem cells. And now, because of this research that we are doing with our collaborator in Vienna, we are also running organoids for developing lungs. But we need to consider always that these are quite embryonic structures. The field itself is still under development. So we are very autocritical in that regard. The organoids can be very helpful, for example, in the examples that we have been putting for this recent work that we did publish together, with regards with the specific cell types that we encounter. Tubular cells within the organoids look much more like the adult cells that we find in the human kidney. But if we are looking for cells that are going to be producing hormones, for example, inside of these mini kidneys, we are still far to be there. So the field itself is evolving, so we are still working on that. But for some specific questions, like the ones that we have been touching here in this meeting for the SARS coronavirus 2, as H2 is highly expressed in proximal tubular cells in the native kidney and also in the proximal tubular cells in the kidney organoid, we are working side by side with both human from patients, cells from patients and from organoids, and we see that the correlation is great. But for other organoids like brain organoids and other organoids that people are developing, the technology itself is still quite new, but so far so good. I mean, we are not the only ones, fortunately. We have a lot of competitors, but now all of us, people that were competing in January, we are all working together. So I think it's also nice to see that we are putting forces together to put all the organoids for the same question, right? So how to stop?
That's actually, it's great system. I really am very interested, because really you were growing in a 3D structure and we are used to this 2D. And also, I mean, the typical work is made with transformed cells, no? I mean, all the cell lines are cancer cells, so we are working in cancer cells. So it's great to have this system. I'm sure it will be a long way, a lot of things very interesting to be found there. Thank you.
Thanks to you. Thank you.
Okay, I was seeing Jordi smiling because we are seeing networking in action now and synergies, possible synergies that might arouse from here. So that's good to see. It seems that we don't have questions from the audience. What we would like you is to keep on voting for your preferred beautiful science and game changer projects. I would like to, I have a question maybe and it is not so scientifically, but maybe more about innovation to Laia and Dr. Fernandez-Barat and Dr. Magri in relation to the scalability of the production of monoclonal antibodies and the pricing as well in the long run. What could you tell us about that? And how far you might feel from that stages as well.
Okay, I can answer something about this. Well, antibodies are very expensive, so this is a very good question. Are very expensive, but I don't know if in the problem that we have now, the high production could, of course, decrease the price. This is the question that we need to have in mind. No, I'm sure it's different. It's a different problem. There is a pandemic, so I imagine that the price would decrease. Yes. So this is my answer.
Juliana?
Yes, for sure is an expensive drugs. That's clear. I think, and this is something that I try to emphasize in the presentation, that we can do a monoclonal antibody that can work at nanomolar concentration. So that means that you can really use a little. So it's not like the 8 grams that have been used for Trump. So this is the idea. So I think that in along the time we can get much better tools that can be used a much lower concentration. Also, there is somehow, and this is something that is related also with the project that I present, there is the idea that maybe we can also think on therapeutic drugs like monoclonal antibodies that are not going to be administered intravenously, but can be also used as a kind of microbicide that can be used orally or in the upper respiratory tract that can block the enter of the virus. So maybe then you don't need to use as much as you can use. You have to use for if you are administered them intravenously. So I think that there are already possibility and strategy that make them a feasible option, let's see.
Thank you. Well, thank you. And maybe another question to Dr. Mañez, if possible. I understand that you have been using this technique for other purposes in the past. What differences have you seen with SARS-CoV-2 since you have started working with it? That would be the question. And as well, if there is there any actual drug that works with that very innovative techniques? I'm sorry, my ignorance for that.
Well, this is certainly a new approach. We have a very drug that is in a very advanced stage of development. And this drug basically removes also the glycopolymer that we are developing to prevent the gram-negative infections in patients, ICU patients. That drug is ready to enter in phase 1 clinical trial next year. And we are already making all the escalation. We have a Caixa Impulse project and we are working in that. And this drug basically removes also anti-B antibodies. Therefore, is one of the ones that we are testing right now, because as I say, this one is probably will be ready to next year. But anyway, we are also looking for other antibodies in the sense that we are, basically the advantage that we have is that structurally mean this is, we have this platform now with different polymers to which we can attach different antigens, mainly sugar antigens. And anyway, if we really show that what antibodies are really the antibodies responsible, the non-neutralizing antibodies that act as facilitator of the infection. In the glycan array study, we already finished. The study is certainly under analysis by bioinformatics now, because we did over 100 patients admitted in the ICU with severe COVID disease. We are doing the glycan array technology. We already with 700 different antigens, and we are now trying to see if we identify other. Certainly for us, we can probably in 3 months may have new glycopolymers that can also be tested. And as I said, and since we are ready to begin in human clinical trials, I think the safety, of course, here we have large numbers of primates treated. And one of the advantages, and perhaps linking with your previous question, is that the technology is a very cheap technology because it's a synthetic molecule, and even that the synthesis of sugars are not really easy. But compared, for instance, with all the other technology, our technology, for instance, will be even cheaper than the use of any generic antibiotic, for instance, used to treat infections. Therefore, it's a very cheap technology, which means if we are able to show that we can generate immunity and really it works in the prevention or in the treatment of infections, bacterial, and of course here in this case, the one we're interested in, the SARS-CoV-2, our approach will be really relative. It's a very cost-effective approach.
Well, thank you very much. I understand as well. One of the answers before nailed. Well, it is different of producing monoclonal antibodies for, I understand, for different pathologies, but in an emergency situation, I understand that they could be cheaper produced. But I understand that. Yeah, thank you very much. And last question to Juana Diaz. There's a question from the audience, and it is about. And I'm sorry, I received it. The question is about the platform. Can you comment if the nanoparticle that you use to deliver the plasmids, are they FDA or EMA approved already?
Yeah, they are nanoparticles to deliver RNA, okay? Because what we deliver is RNA based. Yes, they are. It's actually a commercial one. This is not a I mean, we chose a commercial one to be, to make things faster. So it's named JetPay. So it's no problem at all to say it.
Okay. This was a question from Annabelle Sanz. So, well, thank you again. I think that we have to keep on moving. So thank you again for your very interesting and clear presentations. And Jordi, over to you.
Yes, thank you very much, Ignacio. Thank you to all the panelists. I think that voting is ongoing. Please scroll down the screen to see all the different projects and be able to vote. I think we'll have difficulties at the end of the day in choosing both recognitions of beautiful science and game changers. I think we have seen, I would vote for the for the 10 projects that we have seen already for both categories. So it's going to be a very difficult selection. I can tell you that we are more than 200 people connected. So we are 200 people following this seminar. So thank you for your patience. A special thanks to the Houston Control Center in Girona. I mean, half of the team is there and the TV producer is there. So as you can see, we are finding a second job as TV producers and anchor people in TV programs like this one. But we do enjoy very much. So, well, now it's time for some commercials, for a bio break, some ads. Let's finish voting the Beautiful Science Game Changer project. Let's also see how the voting for the first vaccine to be so chimpadenovirus platform is doing good, although the potential, I mean, all the hiccups in terms of safety during the clinical trials. Please, Moderna team, BioNTech team, Chinese team, Novavax team, or go ahead. I mean, no, it's not lost. It is not lost yet, the match. And so, BioBreak, let's reconvene. Please, I mean, the 200 of us, I know your names. Don't leave the program. Don't go to the next Teams or the next Zoom. Tell them that you're late, that you're attending a much more interesting seminar this afternoon, this evening. And let's reconvene in 55, I mean, at 4:55, like in 5 minutes, right?
Okay.
BioBreak, 5 minutes. I want to see everyone here now immediately after the BioBreak. Thank you. Okay, so
hola.
Welcome everyone. Welcome everyone again. So, as I said before, we had more than 200 people connected following this seminar, so no pressure for the panelists. So, I mean, there is a great expectation in getting to know your beautiful science and your game-changing research projects. We have seen before, and this is quite interesting and was one of the main goals of this webinar, connections and cross-pollination between projects that actually didn't know each other before this panel. So we are lots of groups, researchers, academics, scientists working all together in COVID, all coming from different backgrounds, different universities, different hospitals. And I think it's a great experience that we are working all together today, trying to push forward the research for finding a cure, finding a vaccine, finding new solutions, diagnostics, etc. So today, in this panel, we have the vaccines panel, and the chairman of this panel is going to be Montse Davant, Head of International Relations and Advisor for Research of the Secretariat of Universities of the Research, Enterprise and Research Ministry of the Generalitat de Catalunya. Montse, the floor is yours.
Thank you, Jordi. It's a pleasure to chair this session today on behalf of the General Director for Research, the Ministry of Business and Knowledge. And also, on his behalf, I'd like to recall very quickly a call that is open, which is called Resilience for Growth. And it's open to all fields and to all issues related to the global and local impact for COVID-19. This is addressed to universities, circular research centers, and outstanding large infrastructures. So thank you for allowing me just to mention this open call that can be, I think that it might be of interest to the groups presenting today and also to the attendees to this session. So now, without further ado, I'd like to introduce next session speakers. So we'll start with Dr. Jorge Carrillo from IRSI Caixa, Dr. Cristina Fornaguera from Institut Químic de Sarrià, Dr. Julia Prado from IRSI Caixa, Institut de Recerca Germans Trias i Pujol, Dr. Narcís Saudí from Vall d'Hebron Institut de Recerca, VIR, and Dr. Pere Jaume Cardona from Institut de Recerca Germans Trias i Pujol. So before that, also let me recall you that your participation is very important. Help us choose the winner of the 2 awards, starting now, Most Beautiful Science and the Game Changer work, and you should do it according to your colleague pitches, but also what you see beyond the awards. And also help us guess which vaccine will be first approved by the European Medicines Agency. So, Dr. Carrillo, the floor is yours. Thank you.
Thank you very much. Hello, everybody, and good afternoon. I would like to thank to the organizer for giving me the opportunity to present our result here. I would like to say that we are working the development of a vaccine against the SARS-CoV-2. But we think that in parallel to this work that we are doing, it's very, very important to understand how the immune system is working against this virus, because all this information can give us important tool to refine the vaccine that we are developing. With this aim, we have performed a characterization of the humoral response in COVID-19 patient, and as you can see here in the graph that I'm showing, we developed a set of ELISA that allow us to analyze the humoral response against different antigens from this virus. What we observed was that we can detect all main three isotopes, the IgM, the IgG, and the IgA, targeting different antigens from this virus in almost all the patients that we analyze. But for this first analysis, we can identify that the S2 and also the RBD show some advantage in this serology test, because they show the high sensitivity to identify the patients that were infected with this virus. Something that was surprised, at least for me at this point, was that we can detect these three isotopes from very early after infection. As you know, the humoral response usually work with a first response, mainly dominated by the IgM, and after 1 week, 2 weeks, we can start to detecting IgG in the area. Well, this is not happening here with this virus. We are detecting from the very beginning all these three isotopes, IgM, IgG, and IgA. Can you go to the next slide, please? And this is particularly easy to see here with this correlation analysis, where you can see that there is a strong correlation between all the isotype against all the antigen that we have evaluated in this ELISA. And also there is also a strong association between the antigen that are targeted by these antibodies. You can see in the panel on the right that there is a nice correlation between in the IgM response against the different subunit of the spike, indicating that the IgM response is a polyclonal response that are targeting the S2, the S1, and also the RBD region in this subunit. But something that was interesting was that when we analyze the IgG and the IgA response, we can identify some patients that show a bias to the S2 region. So this result also confirms that the S2 region is a nice antigen to include in this serology test, because they are increasing the sensitivity of this assay. Something also that was very interesting was the nice correlation that we can observe for IgM, IgG, and IgA when we analyze the reactivity against the S1 subdomain of the spike and the RBD, indicating that most of the antibodies that are targeting this N-terminal region of the spike are actually targeting the RBD region. As you know, most of the neutralizing antibodies that have been identified today are targeting the region, because the region is the responsible to bind to the AC2 receptor on the surface of the cells. So as you can see here, IgM, IgG, and IgA are recognizing the region. By this, we want also to analyze. Could you please go to the next slide? According to the result, we were interested also to analyze what was the dynamic in appearance of the antibodies that are neutralizing the virus, to analyze what we perform a neutralization assay, and we can observe that most of the COVID patients are developing neutralizing antibody very early after infection. About 10 days after symptom onset, we can detect in most of the patients neutralizing antibodies. And something also that was very, very interesting for us, because as I mentioned at the beginning, we are interested in the development of a vaccine against the SARS-CoV-2. Some researchers show that those patients that are suffering from a severe form of this disease, they are developing high titer of total antibody against the spike or other antigen, and also high titer of neutralizing antibodies. And from this result, from this work, the researchers suggest that maybe the human response can play a role in this severity 4 of the disease. So we perform a correlation analysis, and our results show that there is not a clear association between the titer of neutralizing or total antibody against different antigens from the virus and the severity of the disease.
You have like 10 seconds, like 20
Yeah.
Sorry about that.
So could you please go to the last? In this last slide, you can see the conclusion of this work that I have mentioned during the presentation. And finally, I would like to thank all the people that are forming part of this consortia, the CEVIC consortia. Of course, I would like to thank to our sponsors and all the people that are contributing to our research through the Yo Me Corono crowdfunding initiative. Thank you very much.
Thank you, Jorge. We go really fast to Cristina Fornaguera, and just let me tell you that we are like 30 minutes late, but it's fine. We will take our time to listen to all our panelists. So please, Cristina.
Okay, thank you very much. So good afternoon. Today I will present you our COVID nanovax project. Next, please. Which is a project that was funded by the express goal of the Instituto de Salud Carlos III, and as you will see, we are a group of materials engineering here at IQS, Institute of Chemistry of Sarriá in Barcelona, and this is a project in collaboration with the University of Cali. You will see that our point of knowledge or expertise is very different from all other presentations, but I would like during these 5 minutes to convince you that this is an outstanding research and that will become a game-changer technology. Why? Because it's related with nanomedicine. Next, please. And why nanomedicine? It could be a game-changer technology. First, because it can improve the safety of the treatments. I will show you that we are trying to develop, as it is written in here, an mRNA vaccine based on polymeric nanoparticles to prevent this COVID-19 infection. Using this mRNA as an antigen and using targeted nanoparticles, you will see how effective this treatment can be. Next, please. But let's take a look deeper in the project. As you can see here, and as I just told you, we are developing a vaccine which is based on an antigenic mRNA, of course encoding for the SARS-CoV-2 spike protein, that will be encapsulated inside our proprietary polymers. In fact, our expertise is in the design of biomaterials, so we have a library of oligopeptide and modified polybeta-amino esters that, thanks to their cationic charges, can complex the negatively charged nucleic acids and form these COVID nanovax. These particles will be targeted specifically to dendritic cells, so we can achieve that they selectively recognize these cells to achieve nanoparticle endocytosis and make that mRNA is expressed only in the cells to achieve antigen presentation and this antigen-specific immune response. We want to generate the immunological memory required to protect patients against the SARS-CoV-2 infection. We have designed the following methodology to achieve this project. First, the mRNA antigen needs to be selected, as well as the most appropriate polybeta-amino ester from our library. Then we need to formulate our nanoparticles to further demonstrate their proof of concept in vitro assays and finally perform preclinical and clinical development. So the main milestones of the project to be achieved are the first one, to select which are the antigens of election. We have selected B anti-epitopes to achieve an overall response, and this has been done by using the S1 fraction of the spike protein. Then we have selected, as I just told you, the proprietary polymers that selectively transfect dendritic cells to achieve the antigen presentation and the memory generation. So the final goal of our project, as all of you, is the COVID-19 prevention. Now I would like to share with you our preliminary results that we have already. We have demonstrated that we are able to formulate these nanoparticles and that they are appropriate for the parenteral administration. They have sizes below 200 nanometers, a low polydispersity, and a cationic surface charge that will enable their interaction with the cells. And one of the most important points is that this high encapsulation efficiency that you see in this slide. We achieved that more than 80% of the mRNA is effective.
So in terms of production process, we will we will be able to design a highly efficient biomanufacturing process. We have also already demonstrated that we are able to in vitro transfect very efficiently dendritic cells, among other antigen-presenting cells, by using a reporter gene.
I interrupt you 1 minute.
Yes, sure. In vivo studies, next please, you can see here that all particles are exclusively accumulated in the spleen, and specifically, next, in the dendritic, in the antigen-presenting cells of the spleen. Next, please. Next. So, at which point are we in our project right now? We are performing the preclinical study. In fact, we have already immunized mice, and we are waiting for the results. And we expect that in the next year, we will be able to confirm the safety and generation of neutralizing antibodies in clinical studies. Before finishing, I would like to acknowledge Biocat for giving us the opportunity to present our work in this seminar, and all of you for your attention. Thanks.
Well, thank you. Perfect timing. Now we have Julia Prado from Caixa and IGTP. Thank you.
Thank you. First, I would like to thank the committee for the Chasing COVID session to select this work for presentation in this vaccine session. And also, I would like to acknowledge all of our collaborations across the different institutes and our funding that has this project is currently supported by the Department of Health in Catalonia and also the Carlos III Institute of Health. So our project is based on the idea that current COV-2 immunoanalysis and vaccine development are heavily biased by numerous potential erroneous assumptions, just because we don't know yet enough about the functional correlates of protection and their cross-reactivity between stress. So our project basically aimed to test with high resolution T-cell immunity in an unbiased manner against the entire coronavirus 2 proteome. We can do that because we count with the existing availability of unique and extensive cores of coronavirus 2 infected and uninfected individuals. Please go to the, thank you. And can leverage with techniques of vaccines developed in our lab for vaccine design. In fact, our team unifies medical staff at the forefront of medical care for COVID-19 patients, well-established immunologists with experts in antiviral immunity, and our state-of-the-art research centers that has the necessary infrastructure to conduct the planned analysis. So we have approached this project through a ex vivo high-resolution immunomapping of the whole coronavirus 2 proteome. And since April, and the paper was recently published in August 2020, we have been able to design more than 2,800 peptides that they will cover the whole coronavirus 2 proteome. That this is a challenge for the scientific community to understand immunology behind 30,000 base pair virus. So we are currently running this high-resolution immunomapping, and we are seeing that not all the response are spiked. So what else we need to protect against coronavirus? At the same time, we are profiling single cells for protective responses by improved proprietary boosted flow cytometry and single cell transcriptomics. And this is not possible. I think this is something to highlight in this project, that we have an existing sample base for access to units and large longitudinal cohorts. Is the ProEpic cohort study that will be presented by Dr. Concha Vilan in session 4. That will allow us to have a big database and integrative analysis. That means higher number of individuals, identification of rare phenotypes, and a follow-up, not only the first wave of infection, but also the second wave of infection. And last but not least, I would like you to convince you why this project has a high impact, not only in the scientific aspect, because we think that we can contribute to the definition of protective immunity against coronavirus 2, but in terms of innovation through the development of diagnostic tools to address coronavirus 2 immunity across cohorts, and also reproductively testing for vaccine prototypes, not only one vaccine prototype, but all the vaccine prototypes. We will ask whether we are enhancing and inducing T-cell responses. Essentially, these new vaccine prototypes that we will be able to inform with our studies will have the aim to achieve a high degree of protection in the population. So why you should vote for our project? Because we think we can accelerate science and innovation through the second generation of a coronavirus 2 vaccine prototypes to optimize protection in the population. And also because we believe that we can contribute to a paradigm shift that it will be to characterize population herd immunity, not only by the limited information that we are taking for the serological tests, but also supplementing or replacing it. Thanks for your time, and that's all.
Oh, sorry, I jumped when you were just finishing. So thank you for being so brief also, and thank you for being so clear. So let's recall that all our colleagues here are competing for beautiful science and game changers. So please take their presentations in mind and scroll down. Remember to scroll down to vote all the possible modalities that are available. So sorry, and don't hate me for cutting you, sorry, because of that. Now we jump to the next one, Narcisa Saoui from Valdebron, please.
Yes, first let's confirm that the microphone is working. Is the audio working?
Yes.
Is it working? Yeah.
Yes.
So thanks. So first, thanks to the organizers to invite us to present our project, COVID-Vac, a fusion inhibition vaccine design model for SARS-CoV vaccine. And let's take 30 seconds to thank the participants. That's Wei, that's making a terrific job in the lab, and Dr. Joseph, the PI, and also collaborators Josep, Francesca, and Paco from the liver disease in our institute, Dr. Oliva from Pompeu Fabra, and Rogier and Quinten from Amsterdam. So since last December, the SARS-CoV virus is all over, just causing the COVID pandemic. A lot has been said about this virus. I only want to mention that it's an enveloped virus. This is important because our vaccine is based on that, the interaction with the membrane. Since then, more than 200 vaccine candidates have been developed, making use of all kinds of vaccine models, protein subunits, inactivated viruses, recombinant viral vectors, RNA, DNA, and most of them are directed against the either full virus, full spike, S1, or the receptor binding domain only. But our candidate is targeted against the S2 subdomain. And as a kind of summary and making a bit of a spoiler of the vaccine, let's say that our vaccine model is based on a chimeric human papillomavirus-like particle. The immunogen we use is heptad repeat 2 and 1 from the S2 subunit. And the idea is to elicit antibodies against this region that binds to R2 and heptad repeat 2 and heptad repeat 1 so that it prevents fusion. Next, please. Why we have selected S2 as a target and HR2 as the first candidate? As I mentioned before, SARS-CoV-2 is an enveloped virus and it has a membrane. And so that the infection goes over, we need that this membrane of the virus enters in contact with the membrane of the cell and they fuse and the viral contents can go into the cell. So to make this, the virus uses type 1 fusion mechanism that's located in the S2 subunit. Here we can see these alpha helix that are the heptad repeats that perform this fusion. Here we can see in yellow that when the receptor binding domain interacts with the cellular receptors, there's a change. This triggers a structural change in the spike protein, and the alpha helix 2, the heptad repeat 2 alpha helix moves to join the 3 heptad repeat 1s to form a 6 helix bundle. This drives cellular and viral members to fuse and form a pore so that nucleocapsid can enter the cytoplasm. The heptad repeat 2 sequence of the fusion core is really conserved sequence, and it's very similar between SARS-CoV-1, 2, MERS, and other coronaviruses. It has been described that peptides mimicking HR2 can be used as a pan-coronavirus interfering peptides. So, and this is what we expect, that the antibodies that we have elicited bind to heptatid repeat 2 or heptatid repeat 1, and this prevents the change in the structure and prevents fusion. Next, please. And how are we going to perform that? As mentioned, we use a successful, safe, and effective vaccine. That's human papillomavirus. That's a virus-like particle. If you synthesize the L1 protein, this L1 protein falls into pentamers, and the pentamers assemble into BLPs. We have inserted the certain residues of the HR2 fusion core into the EF loop of the L1 protein so that it's highly exposed. When the L1, the chimeric L1 falls and assembles into BLPs, we're going to have 660 times exposed this sequence and highly exposed on the surface of the BLP. Next, please. And this is a molecular model of how the HR2 alpha helix is going to be presented in the L1 protein. This model has been performed by our collaborators in Pompeu Fabra and in Amsterdam. And next, several advantages of our vaccine model is that BLPs are more immunogenic than soluble proteins. The vaccine vector we use, the HPV VLP, is a safe vaccine vector, and it's already licensed, and the large-scale production has already been optimized. Also, we make sure that we have a high exposure of the HR2 sequence, 360 times and on the surface of the BLP. Also, L1 protein and human papillomavirus has mucosal tropism, so it can elicit mucosal immunity. And also, the antibodies that perform this fusion interference offer long-term protection as compared to the interfering peptides that I mentioned before. Thanks for your attention.
Thank you very much. And now we move to the last one in this block, Pere Joan Cardona. The floor is yours, Pere.
Hi, thank you very much. And thank you very much for choosing our project. I would like to thank also the Department of Health of the Generalitat de Catalonia for funding this project. I would like to show you, or to to explain a nice story about this project, which is that we are going to do a clinical assay with a TB vaccine. This TB vaccine is called RUTI, that is made by fermented and liposome fragments of M. tuberculosis. And this has been done thanks to the collaboration with IDIAT Jordi Naval, with Dr. Concha Violan and Dr. Pere Turan. And we are also working with Dr. Maria Rosa Sarrias from the Innate Response Lab in the IGTP. So the idea is to use a vaccine, a TB vaccine, against COVID, which at first view you could say, what's this is about? So we have been seeing a nice science about very specific approaches to destroy the virus and to block the receptors. So here we are talking about something very different. Next one, please. So this comes for the need that we are in a sort of run to have a very specific vaccine. So here we give another opportunity to have a sort of broad and specific broad spectrum vaccine against several agents, pathogen agents, included COVID. And this comes from the idea of the BCG vaccine, which is the old vaccine against tuberculosis, has shown that in the countries, that next one that has been is still used, you have a really low impact of COVID, both in incidence and morbidity. So in this case, what we offer is the RUTI vaccine, which has shown that has a very a nicer profile than BCG. BCG is M. bovis attenuated bacillus, so can have some problems, especially in immunosuppressive people. So in this regard, our vaccine is an inactivated one and has a better profile. Next one. So here, next, yes. So the idea here is that, next one, please. So the mechanism of this, why BCG is working, and in fact is now is running a big trial to test that, is that induces this what is known as trinity immunity, which is a sort of different immunity. We usually think about antibodies or T lymphocytes, specific lymphocytes. But in this case, what we do is induce a sort of memory immunity in monocytes. So that the first cells that goes to infectious loci. And what we have seen, has been demonstrated is that the BCG vaccination induced this epigenetic modification. So this modification of histones that allows a quicker response of monocytes once they arrive to the infectious foci. So reducing the load of pathogens. So next one. So here is the design of our clinical trial. So we are going to recruit these 315 professionals that are working in 2 hospitals, in the Hospital Germans de Pujol and Hospital São Paulo. The idea is the ratio is 2 to 1 from routine to placebo. We are going to evaluate the infection and the reduction of infection and morbidity. And also we are going to evaluate in a sub-study the induction of trinity immunity in this sub-study of 60 people, which are now finalizing. So we have validated in a previous trial with non-vaccinated volunteers. We have validated how to perform it, how to perform the analysis of trinity immunity in the lab of Dr. Salias. So this is all. Thank you very much. Next one. So the idea here, the final concept is that RUTI will become a sort of basic wardrobe vaccine for not only for COVID, if the specific vaccine takes some time to be discovered, but also for future pandemics. So in order not to be in such in this sort of in this sort of susceptibility, global susceptibility against a new agent. So this could be a sort of vaccine to be administered at the first time, while more specific vaccine is designed and is efficacious. So thank you very much.
Thank you, Pere Joan, and thank you, Jorge, Cristina, Julia, and Narcís as well for your speeches, for your pitches, and also for being so straight to the point and for trying to compete for these awards. So let's not forget that there are 2 polls ongoing now. We have 1 poll going on all the time, and the polls for our colleagues that will only be open now, I understand, for some minutes. So we, Team Vaccines, need to make our winners, the whole contest winners. So please go ahead and vote for those beautiful projects and game changer projects that we have heard today. So now moving forward, I don't know if there are some questions in the chat, but I would like to ask a couple of general questions for you to answer very quickly, because and understand that it's not a long time we have for this discussion. So, but we see that the array of vaccines that now are under development, there are like 44 on clinical and like 150 something in preclinical. There are different kinds of vaccines. You just showed some of these types in your presentations. These are the mRNA vaccines, the virus-like particle-based vaccines, the protein or proteome vaccines, like you explained also. So we also see that these different kinds of vaccines have strengths and weaknesses, and also strengths and weaknesses linked to the response that they induce, the quality, the durability, and also the number of deliveries, for example. So I want you very quickly to defend or to just answer which is your position on that regard, the strength and in just one word, the strength and the weakness of your proposal. Over the others.
Let me start with our model. The strength is that it uses an already known backbone, that's the human papillomavirus, that's safe and efficacious. And weakness, well, it has quite a bit of downstream processing to get the vaccine.
Okay.
No, go ahead, please.
So on our side, I think a strength is long-term T-cell immunity. So we will challenge the virus and control the virus every time that it comes. Another strength, it comes through cross-reactivity. So we will be protected against other viruses. And weakness, at the moment that we need more basic knowledge, and it will take a little bit longer to generate these second prototypes for a higher protection.
Thank you. Cristina, for example?
Okay, thanks. I think that the main strength of our approximation is the versatility. I mean, even if we have already selected the antigens to be encapsulated, we can adapt our prototype to encapsulate other antigens. And since the particles by themselves would be very similar independently on the mRNA that encode, these changes on the formulation are not highly time consuming. On the other hand, the weakness of our system could be maybe that our proprietary polymers are not yet accepted or validated for human use. But we have performed a lot of tests in other projects that we have before to arrive to clinical studies.
Thank you. Jorge, for example.
Sorry, I have no present data about the vaccine that we are developing right now. I have only showed data about the natural human response in this SARS-CoV-2 infected individual. I would like to highlight for our result and for many other researchers that are doing a similar study that the development of neutralizing antibodies seem not to be a very important handicap in this infection. So most of the individuals are going to develop neutralizing antibodies targeting this RBD region and probably other regions that are not characterized yet in the S2 region. And to me, probably the major point that is going to be different in between the different platforms of the vaccine that are running right now is the durability of the immune response that they are going to generate, and also the safety issue associated with this vaccine.
And I guess in our case, our strengths are that this is a product that is ready to use. We have already done phase 1 and phase 2 clinical trials in TB infected people and TB patients. So it has a very nice profile. We have the scalability done, so this could be really in the market very fast. The other thing is that it could be used as a combination with other specific vaccines. We don't know these new vaccines will have the strength to be efficacious enough. And the third is that it's a broad spectrum vaccine, so it is trained immunity, so this could be also available for other viruses and even other bacteria. On the other hand, the main weakness is that, of course, this is a sort of time-limited vaccine in a way that once there will be a specific one, we should, in a way, retire. But there is also the possibility to merge with other specific vaccines.
So thank you very much. We will move forward, but before, let's recall all the voters that you have to scroll down to see the whole screen. And I'm sure that Jordi will mention about the poll on the vaccines for the EMA. So probably he will recall that. So now, please, vaccine team, go vote. Thank you very much for your participation and the best of lucks for you. Thank you.
Thank you.
Thank you.
Yes. So, okay, so thank you. So again, thanks, Houston Mission Control in Girona specifically. I know, I mean, we all have recycled from project managers or scientists to TV presenters these days. I think we are doing quite well. We still have around 200 people connected. Again, thank you to the team in Girona taking care that everything goes well. Nuria, Marta, and Toti, and the rest of the team. So again, I mean, I hope you see the voting of the first vaccine, which is going to be approved by the European Medicines Agency. I've seen that the Chinese Moderna teams are improving, but still is Oxford winning the day. So yeah. I mean, don't you like adenoviruses? I mean, there are a lot of beautiful adenoviruses there. Adeno 5, CanSino, Adeno 26, Janssen. Oxford is a chimpanzee adenovirus, but human adenoviruses are beautiful. And RNA is beautiful too. So, and the classical virus inactivated vaccine from Sinovac Sinopharm, it's also worth considering. Or the subunit vaccine of Novavax, it's also worth considering. So just saying, not everything is Oxford, right? Okay, so 15 down, 5 to go. Thank you for your patience. We are still 200 people connected. So we have seen 15 projects, excellent science. We have seen vaccines, we have seen therapies, we have seen monoclonal antibodies, we have seen reprofiling, a very relevant area. Now it's time for medical devices, medtech, diagnostics, and population studies. So the last 5 projects of tonight, I was saying afternoon, now it's going more entering into the evening. So the five last projects are in the area of medical devices, diagnostics, and population studies. And the chair of this session is Cristina Vescos, the managing director of EIT Health. Cristina, thank you very much for joining us today.
Thank you, Jordi, for inviting me to chair this last session. As you mentioned, from EIT Health, we are probably the last in the chain of bringing the science to the market because we are dealing with the innovation part, and probably that's the reason we are in the last panel. I can find also other 2 reasons. One is related to the mixed panel that we have, and I think it's one of our mottos that in order to really bring science to the citizens, you need to have really a mix of disciplines and all the stakeholders involved in the value chain. So today in this session we have 2 presentations related to diagnostic devices and then 3 related more to genetic population studies. And probably the third reason I find to be here is bringing a bit of the European approach, and I will be a bit Germanic in the timekeeping as we are running a bit late, I understand. So just for all of you that are in the audience, please be patient. Keep us with us for the next half an hour of this interesting session. So without delay, I would like to present our 5 panelists. So first we'll get Jordi Alonso from the Research Institute of Hospital del Mar. We'll present a study on the impact of mental health of COVID. Second will be Giulio Rosati from the Catalan Institute of Nanoscience and Nanotechnology, who will talk about printing your own COVID-19 sensor as point-of-care device. Afterwards, Nana Rule from Pere Virgili will present also biomarker identification for early diagnosis of COVID-19. On the 4th row, Marta Soler, also from ICN2, will introduce advanced nano biosensors for global point-of-care diagnostic and surveillance. And last but not least, Concepcion Violant from IGTP will introduce a population study on the identification of carrier status and COVID-19 immune response related to progression phenotype. So without any more delay, I will give the word then to Jordi Alonso, Hospital del Mar, who will talk about mental health impact and needs associated with COVID-19, a project called Mind COVID. Jordi, the floor is yours.
Thank you very much, Christina. I hope you can hear me well. So it's my pleasure to be presenting this work on behalf of my colleagues of my own research institute at INIM, at the Hospital del Mar, but also as a part of a collaboration with many other institutions in Spain. As you know, adverse mental health is associated or has been said that it will be the fourth wave of the COVID-19, or even it's called a parallel pandemic. So, but no, can I have go back to the first one first? Thank you. So there is need to measure the real magnitude, to monitor and to know the evolution of the mental health impact in order to accurately inform health authorities and to deploy adequate treatments. So we need rigorous methods and analysis. This is what chronic disease epidemiologists like myself do. And I'm happy to present this study that is funded by the Health Institute Carlos III. Please, can I have this next slide? So to the best of our knowledge, this is one of the largest studies in mental health in Spain. It includes 24 groups in different health institutions, and it covers, say, 6 autonomous communities. It is based on a series of prospective cohort studies with the aim of assessing which is the impact of the disease on mental health. It's also trying to identify risks of the various types that are risk factors and also protective factors that are associated with the onset of mental disorders, but also those that are associated with the persistence of adverse mental health conditions a long time. And it's important that we need to quantify the use of mental health resources that we know are very low in general, and we anticipate they are going to be lower during the pandemic, and identify and mental health needs for care. This project, by mandate, it has to feedback with recommendations to the mental health authorities and the professionals planning and deploying services. Can I have the next one, please? So we are studying 3 types of different populations with the same methods and assessment tools. 10,000 healthcare workers in Spain, 2,000 either COVID patients or close contacts, mostly COVID patients, and then a random sample of 3,500 individuals that are representative of the adult general population. We're using web-based survey platforms for that, and we are measuring a baseline 3- and 6-month follow-up, and obviously we have plans to, not funds, but plans to continue in the future. For those people that are not competent with online technologies, we are using telephone interviews. So I mentioned that there was a number of factors that we are interested in, and these include individual and personal and social, socioeconomic information. It also includes COVID infection exposure status, personal and family and professional. And obviously the center of the study is mental health disorders, post-traumatic stress, panic, depression, anxiety, alcohol use, suicide, and services use, and psychological functioning and health and quality of life. We have already analyzed the data for for the healthcare workers baseline. And unfortunately, these data are now embargoed because they're at the end of the peer review process. But what I can tell you is that prevalences of specific mental disorder is high, including suicidality. Can I have the next one, please, and the last one? We are convinced that our project has the capacity and should contribute to improvement of knowledge. And it should have also eventually an impact on the identification of and meeting of the healthcare needs for mental care. First, I think we are applying rigorous methods and trying to use actionable knowledge or actionable information. Second, we think that we're relatively rapid assessing the health needs of the population. I mean, more rapidly than it has been done in the past, for sure. I think this is something that every one of us is experiencing these moments. And we are also interested not in the immediate impact, which is important, as we already know, but in the maintain in the future and the longer term, because previous studies in previous associated... I'm finishing right now.
Please be ending, thank you.
And so the other important issues that we are thinking of a very important concept, which is vulnerable populations. And I think that finally is we are going to provide a platform that consists not only in the technology, but in the methods and the instruments to evaluate e-health and other interventions that will be used in this during this pandemic. Thank you very much.
Thank you, Jordi, for this introduction of this study. Then we give the floor to Giulio Rosati from ICN2 with his paper on rapid diagnosis of COVID-19 and other viral upper respiratory tract infections through a rapid paper-based device.
Thank you so much. Thank you so much. First of all, I would like to thank Biocat for the opportunity of being here and for the very nice format of this virtual seminar that I find particularly helpful for our research. So good afternoon to you all, and I'm Giulio Rosati, a researcher in the Nanobioelectronics and Biosensors group led by ICREA professor Arben Mercosi at ICN2. And today I will introduce you to a new paradigm to spread COVID-19 biosensing and to make it effective, fast, and possible at the point of care, thanks also to a CSIC project received by our group. You already know why we are here. At the beginning of this year, we experienced what many researchers have advised that would have happened for years. A pandemic originated in China and rapidly exported in our countries, thanks to our interconnected and globalized society. Next, please. The infection detection is paramount to contain a pandemic, as you know. However, so far, we have been able to introduce just two methods, each one with relevant drawbacks, unfortunately. PCR is a standard method for the virus detection in nasopharyngeal samples. However, it's pretty expensive. It takes 24 to 48 hours to give a result, and it requires qualified personnel in laboratories. On the other side, serological rapid tests take 15 minutes, so they are pretty fast at the point of care with blood samples, but only check for the subject's response to the virus, eventually developed several days after infection, or in many cases, they cannot distinguish the infection from a normal cold. So now, let me present you the device we are developing to face these issues for a rapid and point-of-care solution to the detection. But this is not only another sensor on the shelf. What we really propose is not the sensor, but the platform with a print your sensor capability. So in fact, our sensors are fabricated by inkjet printing with low-cost equipment, an office printer, and automated procedures, which could be performed by unskilled personnel. Our goal is to distribute the production capability instead of selling the sensors, making every country, overall those with low resources, autonomous in their fabrication and making the sensors available very fast for those who need. In particular, our platform has a fixed cost lying under 1,000 euros, is pretty cheap, and the variable costs are under 0.5 cents per sensor. The platform allows now fabricating up to 5 sensors per minute for small volumes, but much higher rate is possible with high volume production. Our sensors are read by smartphone with a commercial app, and they are easily functionalized for SARS-CoV-2 spike protein detection, so for the whole virus detection, in less than 10 minutes. And regarding the applications, next slide, please. The application in the clinics are, I think, pretty obvious with decentralized mass production and massive testing, but also with important consequences for the production in developing countries, for this and for future pandemics. While less obvious but equally important applications are related to the monitoring of the territory, for example, with sewage water monitoring for target and precision containment actions, for which we are writing a project proposal for the Agaur Pandemies 2020 call here in Catalonia. And this is a very important application because it covers the gap that sometimes we are seeing between science and politics, economy. So, well, from my side, that's all. And for further information or any question, feel free to contact us by email. And of course, thanks for your kind attention.
Thank you, Julio. Perfect timing. So you even finished earlier than planned. So very interesting presentation. Let's then move to Anna Rull from Pere Virgili, who will present her paper circulating and cellular prognostic biomarkers for COVID-19 progression in patients with SARS-CoV-2 infection using multi-omic science. Anna, the floor is yours.
Good afternoon, and thank you for giving us the opportunity in the next 5 minutes to tell about our project COVIDomics. So, as you could see in the next slide, please, COVIDomics aim to improve the knowledge regarding the immunopathology of COVID disease and to predict, to search for predictive markers of COVID severity. And to accomplish this objective, we are creating a COVID patients cohort, and we are classifying the patients based on their clinical evolution. We believe that our results will help us to identify potential therapeutic targets for this global pandemic. So in the next slide, you can see that both post-COVID positive patients and COVID positive medical staff from our hospital that consent to participate in the study are included at the moment of diagnosis, and we are follow up them after 4 to 8 weeks of infection. Then we are classifying these patients in 4 groups depending on their evolution as asymptomatic, low-grade, moderate, and severe. And also we are recording information about previous comorbidities such as obesity and diabetes to determine their association to COVID severity and clinical prognosis. From March to June, we recruited more than 170 patients, and because it's an open cohort of a study, now we are around 300 of patients. At this moment, we are performing a multi-omic approach, and for this task, we contact to María José Buzón and Meritxell Genescà from Vall d'Hebron Institute de Recerca, and also with Ezequiel Ruiz Mateos and Alicia Gutiérrez from IBIS Sevilla. And both groups send us some of their samples to include in our omics analysis. We perform analysis of 400 samples, including both baseline and follow-up time points. Our objectives are to understand what makes the different clinical progression happen with the study of proteome, and also to answer the questions what has happened or what is happening with the study of the metabolome, which includes the changes of metabolites and also lipids. We have started with interpretation of proteomics data, and in the next weeks we think that we will be able to start interpretation of metabolomics and also lipidomics. At the same time, we start a collaboration with the Cancer Epigenetics Group from Institut de Recerca contra la Leucèmia, the Josep Carreras. They contact us as a result of a quick COVID network meeting, and they will help us to answer the other question, what can happen with the genomics and epigenomics study? We send them around 130 genomic DNA samples, and we are now waiting for the results. The integrative analysis of all these data set obtained from the multi-omic approaches will be completed with the measurement of specific circulating inflammatory markers. This task will be done by two different groups of our institution. The AMET group will determine succinate and succinate-related molecules as an excellent predictive biomarkers of inflammation status. And the other group will perform a multiplex analysis, and is especially interested in the study of the association between obesity and COVID severity. And for the next month, we will also try to isolate different cellular subtypes that we know that are crucial in the immune response. So in this last slide, we show you the research impact that we hope to obtain with this project that we are starting analyzing the data. Because we are creating a well-defined patient cohort with detailed clinical data, integrative multi-omic signatures, circulating concentrations of different inflammatory markers, we anticipate the identification of predictive biomarkers of COVID severity, but also the identification of unexpected and or unknown key molecules in the immunopathogenesis of COVID disease. Thus, we believe that the obtained results will be addressed to attend the necessities of the last end users, that are the patients, but also the health system, allowing the physicians for a rapid diagnosis of their patients, anticipating to the disease evolution. So this is the information that I tell you about our project, but please, if you have more interest, you could contact with us, and it's okay. Thank you for the attendance.
Thank you, Anna. We have passed in the middle of our session, and I think you could all agree that we are seeing very good examples of beautiful science and game changers. So please note that for the voting that will start again at the end of the session. And let's move then to the next speaker, who is Maria Soler from ICN2, who will talk about the COMBAT project. It's also a biosensor for diagnostic and surveillance. Thank you, Maria.
Hi, hello. So I will talk about this COMBAT project. Probably most of you have heard about. It's a European project that started in early March, just when the pandemic reached Europe. And the project is a collaboration with the University of Barcelona, the University of Marseille in France, and also the Institute for Infectious Diseases in Italy. But also during this time, we have fostered some local collaborations with the Vall d'Hebron and the Clinic Hospital here in Barcelona, with the University of Valencia and the CMB Madrid. So all in order to accelerate the validation of our technology for the diagnostics. So if you can play the video, what we are trying to do in short is to adapt one of our pioneering nanotechnology for biosensors that will be able to diagnose the COVID infection fast and in a reliable way. And it will be, of course, applied for patients, for human patients, but it's our idea also to adapt it for the monitoring of coronaviruses in bats, for example, to control the evolution of these viruses and prevent for future outbreaks. So the next slide, please. So our nanophotonic biosensors exploit the light properties of the nanoscale, so we are able to perform label-free and real-time analysis of any biomolecule in general, also viruses and bacteria. We fabricate the sensors here in Barcelona at large scale, so they are cheap, and we can use them as a disposable cartridges in portable devices that can be operated at the point of care. So, for example, in primary care centers or pharmacies. And also thanks to our collaborators, we have access to high quality material like SARS-CoV-2 samples inactivated, specific antibodies and viral antigens, and also human clinical samples and animal samples for the validation. So next, please. So the main advantage of our technology is that it's very versatile and is super sensitive. So what we want to do is develop one platform to perform 3 types of assays. One is the direct virus assay. So we are able to detect the intact viruses in saliva samples in just 15-20-minute samples. And also we are able to provide the viral load in the patient. So it's a kind of prognosis, not only diagnosis, yes or no, but prognosis to closely monitor the patient for possible complications. We also have an additional confirmatory test based on genomic analysis. So we are able to identify the viral RNA without the need of PCR, the amplification. So the total assay time will be around 30 minutes. And we can also multiplex. So we could distinguish between COVID, flu, common cold, or different type of viruses. And plus, besides the combat project itself, we are also developing the serological assay with our biosensors, because we can achieve very sensitive detection of antibodies and also to quantify them. So rather than a yes, no to the presence of antibodies, we can quantify them. So we believe this is very useful for vaccine evaluation and development and to assess the immunity response in patients. So next slide, please. So I would like to show our first results. For example, for the direct virus detection, we are already with very good sensitivities. We are detecting less than 100 viral particles per milliliter, taking into account that the minimum levels found in patients through PCR are between 10 to 3 to 10 to 5, usually around 10 to 7. We are well below the levels in patients. We are now testing the detection in saliva samples, and we expect to validate the sensor in the following months. And the next, please. So for the serological assay is the one we have more advanced. We are already in clinical validation of the sensor. In this preliminary study that I show here, it's made with control and patient samples. We were perfectly able to discriminate between negative and positive COVID patients, and also to relate the amount of antibodies to the severity of the infection in the patients. So next, please. So in conclusion, I would like to say that we believe that we are on the way to provide a portable and rapid device for COVID-19 screening. We offer quantitative detection at the point of care with easy and non-invasive sample obtaination. Our approach is a multi-target and global diagnostics that can be used for any type of viruses. And yeah, we want to prevent disease spread among human patients, but we want also to prevent future outbreaks by monitoring the coronavirus evolution in reservoir animals. And that's it. Thanks for your attention.
Thank you, Maria. So we've seen already in the 3 projects related to diagnostics, so rapid, portable, cheap, accurate, and even print it yourself devices, definitely that can be game changers in this fight against COVID. The last presentation goes to Concepción Violan from Germán Trias y Pujol, and she will present a population study related to the identification of carrier status of SARS-CoV-2 immune response related to progression phenotype. So, Concepción, go ahead. Concepción, I think you are on mute. You need to unmute yourself.
Okay, sorry. I will start again. And then, good afternoon. First of all, I will give thanks to the institutions to organizing this event and allow us to present the population study, identification of carrier status and SARS-CoV-2 immune responses related to the progression phenotype. The name and acronym is PROGETIV-19. This study is led by the researchers from IGTP, Jordi Naval and Caixa. The names of the researchers is Dr. Pere Toran, Julia Garcia Prado, and Eva Martinez Cáceres. And also, this project are involved the primary health centers and the Germans Trias i Pujol Hospital of Metropolitana North Area of Barcelona. This study is funded by the Department of Health of Generalitat de Catalunya with the collaboration with Biocat. Next, please. One of the key questions is the people who have been infected with SARS-CoV-2, whether symptomatic or asymptomatic, develop permanent immunity to the virus. The answer to this question is crucial for epidemiologists, policymakers, professionals in the health services, and the society in large. In essence, this project prioritizes now is the how best control and predict the courses of pandemic. By performing the long-term immunological studies of the population, we will be able 3 points. The first is characterize the kinetics of SARS-CoV-2 antibodies with ELISA or another flu reagents for the detection antibodies against SARS-CoV-2. Also, identify the individuals with a strong immune response against SARS-CoV-2 and determining the duration of the humoral and cellular responses to the virus. And third, determine the role of innate and adaptive immunity, plasma and blood, with the TB lymphocytes and cytokines against the SARS-CoV-2. The aim of this study is to analyze the differential characteristics of the immune response against SARS-CoV-2 associated with the disease outcome in asymptomatic versus asymptomatic carriers in long-term course. Next, please. First one, how we developed this project. We designed a prospective core with 4 cores of health workers and 1 core of general population. The first core of health workers was 1 for prevalent professionals. That means the professionals is infected in the first wave. The second is the professionals is infected in the second wave, we mean the sick professionals. And the symptomatic incident general population is the third course of health professionals and exposed population with a high or low risk scores. The 5th core is the individuals of general population is infected in the second wave. The variables of this study was the diagnosis and comorbidity the people has, sex, hospital admissions, and COVID symptoms. We decided to do the RT-PCR performed at the inclusion period of the study and 14 days after inclusion. Also, we measure the antibodies analyzed of the inclusion period and in different periods of time of the first RT-PCR test. And also, we determine the T cells and cytokines analyzed in different periods of time after the 5th PCR test. The study is follow up during 1 year. And the next, please. The study, as before, is developed in the Metropolitana North area and included 600 population, and the sample of the study is 600 health professionals and 150 general population. Next one, please. What is the impact of this project? First, the scientific and innovation and social. We do the accurate assessment of the antibody response during the pandemic in the first and the second wave. And we have an important epidemiological data on exposure of SARS-CoV-2 and herd immunity. Also, we're obtaining a kinetics of the SARS-CoV-2 immunity, humoral and cellular, and do important epidemiological data on exposures of SARS-CoV-2 and herd immunity with the big numbers. We do the acceleration of knowledge because Project Core 19 is a basis for more than 6 COVID projects basic to translational. And the Dr. Julia Prado said, and a strong collaboration with the high resolution of COV2 specific T cells immunity to drive broad coronavirus vaccine development project will allow to identify the cellular immunity between the different cohorts and complement the information of development of vaccine. This is the paradigm shift because words are turned into reality. The translational research between clinical, basic science, and population research. And finally, I would like to thank to the health professionals are involved in this project who are, I would like to thank to the health professionals and general population who are involved in this project. Thank you for your attention.
Excellent presentations, Concepcion. So thank you very much, all the 5 panelists. So we have closed the round of presentation and we will open briefly the round of questions and answers. While we allow questions coming from the chat, I'll remind you again that the vote is still open for the beautiful science and game changer. And I know we cannot vote for vaccines in this panel, but please scroll down because I think we've seen very interesting cases both in the part of epidemiological studies. So really understanding what is the antibody response and the impact of mental health of the pandemic. And in the part of biosensors, how to make really diagnostic tools available for the populations in need of them. No? So while you are voting and preparing your questions, I'm going to throw a first question to the panel, and it's a bit of also on our own interest. So from EIT Health, what we've seen in this pandemic is that this translation from science to innovation and to the community has accelerated immensely. No, we've seen thousands of publications per week. Hundreds of webinars and so on. At the EIT Health, we had to launch a call for COVID actions, combining rapid response projects and to rescue also startups in the middle of raising funds of 40 million euros in just 6 weeks. And we've seen that all this science ecosystem has accelerated. But apart from speed, I wanted to ask you, and maybe it's open to all of you, what has changed, and not only just the topic of your research, in the way you develop and you do your science since the COVID outbreak?
Certainly, it has changed the teleworking has allowed collaboration with many different colleagues in many different areas. The other thing I wanted to mention that I think it's we are a little bit rushed, and probably the rush is totally is a challenge for the paradigm that we were doing science. I'm not saying we should not be very quick, but I think it's a challenging situation.
From my side, my opinion, I think that we are, with this pandemic in particular, working in the development of biosensors for diagnostics or monitoring. I think we are all more conscious of why we are here and of what we have to do and of what we do. But what we do, we all do in the people attending to this seminar. These things can save life and should save life. It's a big motivation from my side, but and these allow us not to get lost in science for love of science. But at the same time, it's a kind of tremendous responsibility. So if we have to talk about how we feel or what changed, it changed that we are focused more to come out with something that works rather than publishing in Nature. Then if we can do both, better. But now, at least from my side, I think we are more conscious of the importance of what we are doing.
Yeah, I would like to remark what Jordi said, that regarding the collaborations. So from our experience before, fostering collaboration was kind of difficult. It had to go contact to contact. And now with all these webinars, I mean, in our project, we have started like a lot of collaboration with not only in Catalonia, but in Spain and even Europe. We have been applying for multiple projects. And those collaborations are kind collaborations. I mean, nobody is asking for reward, but just helping to develop faster and validating faster the devices, so we can really push the innovation towards the end, right?
Yes, the
Thank
common
you.
purpose. The
Yes,
common
so
purpose.
so we've seen that I mean the speed is not only the speed, but it's also the purpose of the science that is making a lot of change in the way we do our work. I've seen there are no questions from the audience, and as we are a bit behind schedule, I'm going to give back the floor to Jordi. So one of the things we've seen in this panel is that it's not only about science, it's about translating this whether medtech results and epidemiological studies to really to the market or to the public health policy, and I think it is an important step that we need to do in order really, as Julie was saying, to save lives, which is in the end the purpose of all this speed action around COVID research. So thank you all in the panel, excellent discussion, and just let's give the floor to Jordi Naval.
Thank you, Cristina.
Thank you, Cristina, and thank you to all the panelists. This is as long as the Oscar ceremony, so it's 3 hours, 15 minutes of presentations of a seminar. I'm very happy because you are still there. I mean, more than 200 connected people at any given time, and thank you for your patience. Thank you, of course, to all the 20 participants. It's not, there's going to be one winner of the beautiful science and of the game changer award, but I've seen dozens of beautiful science projects and dozens of game changer awards. But we still have some things to comment before announcing the winners, and it is the result of the vaccine voting. So the audience has spoken, and I think that the winner is clearly the Oxford AstraZeneca vaccine. And I'm very happy because this is the opinion of around, I mean, we have like 100 votes for the vaccine that's going to be first approved by the EMA. And we have invited Dr. José Luis Cabero, and with a long experience in vaccine development. And hello, José Luis, good afternoon, good evening.
Good afternoon.
Thank you for joining us today. And this is like after the Barça matches, this is like the after match conversation. Mm-hmm. So how was the game? I mean, the match was not, I mean, there's a clear winner, and I would like to know, as an expert, your take on your opinion on the Oxford vaccines, on the difficulties of running this kind of trials, and what would be, if you can travel in time 1-2 years from now, how do you see that the population, the world population will be get vaccinated? Is it going to be just one winner? Is it going to be a combination? Maybe, perhaps, just saying a combination of a vaccine with antibodies. So what's your take on this result and on the vaccine scenario right now?
Good. First, Jordi, thank you for inviting me to contribute and to be part of this. I enjoy very much the presentations. I think it's a very good indication of the vibrance that is going around in relation to having ways to tackle this new pandemic that we are living with. Regarding the winner today, of course, it's not a surprise that the AstraZeneca Oxford vaccine is getting a lot of attention and adepts. It was one of the first runners actually to start clinical trials, and the team behind is very experienced, particularly in the chimpanzee adenovector that they are developing, which has the advantage of not having pre-existing immunity in humans as the other adenoviruses that are being used by other vaccines, like adenovirus 5, used by the CanSino company in China, and by the Sputnik vaccine in Russia, or the adenovirus 26 used by Johnson & Johnson, although the last one has much lower pre-existing immunity in humans, which is a much more rare adenovirus. It's very interesting that we are betting to a technology that is new, has been extensively used in clinical trials so far, and is being used in other clinical trials, but has not been reached the market yet. In spite of that, what we have seen regarding efficacy and safety is very promising. We have seen some pauses in some of the trials with the AstraZeneca Oxford vaccine that has been considered not to prevent further development. And in fact, the trials in the UK, in Brazil, in South Africa, and in India with that vaccine are ongoing, and in some geographies probably close to completion. Still is a pause in the US, and there are unofficial information or data going around indicating that they may resume the trials in the US next week. So all in all, this vaccine seems to be in the front runner phase, and it's likely that they will get approval in the EMA, one of the first. I'm not sure that it will be the first because, as everyone knows, the second runner-up, which is Moderna, and Pfizer BioNTech are very, very closely approaching the finishing line, at least for an emergency use authorization. Now, Moderna just announced today that they have concluded the recruitment in their large study, and Pfizer and BioNTech a week ago announced that they started their rolling submission at the EMA in Europe. So it's a tight race between these three ones, with some advantages regarding the distribution in favor of the AstraZeneca Oxford vaccine, because the RNA-based vaccines will need a cold chain that to be distributed, that will require -80 degrees capabilities all the way, which definitely will put some logistical challenges to those of them.
Yeah, I see that we are starting to finalize the counting. Game Changer Awards, but before announcing them, and because this conversation with José Luis is very interesting, I would like to know, José Luis, what your opinion on the classical vaccines, because I mean, it seems like in this case that would be the first option.
It's it's a very interesting question, because I said that it's very interesting to notice that we are betting on novel technologies, while one would like to believe that the more established traditional technologies like inactivated vaccines or recombinant subunit vaccines like the one from Novavax will have further chances of succeeding. And perhaps at the end, they will become more established. They will not be in the front runner. We have 9 vaccines in phase 3 clinical trials, but we have 42 in clinical trials as far as I'm aware of. And that means that we need to be taking a look at the vaccines that are in phase 2 today. Among them, one from Sanofi and using an adjuvant from GSK, a proven adjuvant, that this is a BioNet vaccine as well. So I think still we will see the traditional vaccines probably acquiring an important role, not in the first phase, but an important role. The first phase will probably allow us to have vaccines for at-risk populations and most likely to ameliorate the course of the disease rather than to sterilize and to prevent the transmission of the virus, which will be needed at the end. So still is a way to go with vaccines. Monoclonal antibodies, as you said, will play a role at the very beginning together with the vaccines, but we will see first and second generation vaccines for a while until probably 2 or 3 will become the ones standing out and becoming widely adopted.
Excellent. We may see in the future like combination, for example, starting with an inactivated vaccine and then giving a boost with an RNA, or the other way around. Absolutely. Maybe in the long term, this is probably a combination. And last question, Jose Luis, thank you for your insights. And as I promised to the audience, the aftermatch with you commenting on the results has a lot of value. What about manufacturing and reaching the population when things approval, which we all hope is going to happen in the next few months? Next thing is the vaccines to be available for the general population at the global level. So,
correct. I think this is very interesting, and again is a good proof of what can be done when there is a will and urgency and a sense of urgency. So what we have seen in vaccine development is unprecedented. So the sequence of the SARS-CoV-2 virus was made available the 11th of January 2020. 10 months later, we have what we already have, an enormous amount of people working into different approaches to tackle the pandemic. And then particularly vaccines, we have never seen this. The fastest vaccine that we have seen in history is the mumps vaccine that took 4 years to be developed. Never seen these huge efforts, and among that is to increase manufacturing capacity at risk. So it's a lot of investment coming from different governments and non-profit foundations or organizations, some foundations like the Bill Gates Foundations, and a consortia of public and private partners trying to increase the manufacturing capacity. So we do see possibilities to tackle, and actually we expect to have a significant number of doses early next year and during the course of next year to target risk populations. But we need to manage expectations. The vaccine will not be the magical bullet to manage alone the pandemic in the near and medium term.
Yeah, and final comment. I mean, from your side that are actually working in the vaccine field, and also from my knowledge, and I think you would agree with this, I don't think we are compromising the safety of the study participants with the speed. It's not the speed at the expense of safety. I think that all the trials are keeping very strict pharmacovigilance and safety measures to protect the study participants. and really it has a lot of merit to be able to deploy these complex clinical trials and at the same time keeping everybody safe and really studying all the potential adverse events connected with the eventual use of the vaccine. We have seen several trials being paused several times just because one case among the 15,000 showed some kind of safety signal, right?
Yeah, I think it's a very, very legitimate comment, and I think we have seen that all the sponsors are taking the care that is needed to watch safety. But we need to remember that probably the first authorizations will be for emergency use, and the trials will continue in order to increase the safety information. Still, one thing that will be needed to be looked after is the cases that are very rare, but they do exist or they have occurred in some vaccines, which is the vaccine-dependent disease enhancements or antibody-dependent enhancement, which is the fact that some pre-existing immunity or the production of non-neutralizing antibodies may contribute to enhance the disease. And in order to see this, we will need to see much larger exposure to individuals. But this is a matter of time and exposure, not a matter of neglecting this at this point.
Excellent, José Luis. Thank you very much for your time. Thank you very much for your wise comments. So the moment of truth has arrived.
Thank you.
Are you prepared? Are you prepared to know the winners of the? First of all, we'll announce the winner of the Beautiful Science Award of the Chasing COVID webinar, which goes to Nuria Montserrat from IBEC for her project, Engineering Mini Organs to Understand and Target SARS-CoV-2 Infection. So Nuria, congratulations, and thank you very much for your contribution.
Thank you. Thank you very much for the organization of this wonderful meeting, for letting us space for explaining the science that we do all together. And this is not the merit of only part of my institution. So I'm very thankful for this moment. Thank you. Thank you very much. And for all the attendees and to everybody. This is really nice. Thank you.
Okay, I don't know, we'll have kind of an animation to celebrate the prize. The Girona Houston control did.
With this Zoom, I don't know. I think that some people can even put some stuff back.
Okay, so thank you, Nuria. I think it's a beautiful project. It's a beautiful science. It's really creative, innovative, and not only for SARS-CoV-2, but for many other diseases and viral diseases. I mean, this concept of organoids as models, quick models, where to test vaccines, development, antibodies, etc., is really very it's going to bear many fruits in the future. Thank you very much. And the next and the final prize award for the game changer. I think we are all in agreement that besides vaccines, what we need right now are quick, affordable, reliable tests, diagnostic tests. So the winner of the game changer is Maria Soler from ICN2 for his project Convat, that was in particular featured in one of the latest issues of Nature Biotechnology as one of the game changers diagnostic tests that can change the course of the disease in the following months. So congratulations, Maria, and congratulations to your team.
Yeah, thank you. And yeah, that's it. It's a big team and we are working very hard for this. So Yeah, thank you very much. Thank you for the opportunity for sharing the research with such amount of people. And yeah, we are open still for collaboration, for investment, for anything. So if you just want to know more about the project, just text, send an email, phone call, whatever, and we'll be here. So, and yeah, thank you very much.
So thank you, everybody. And thank you for your patience, and thank you for being here. I think that we have seen excellent science, beautiful science, game changers. And what makes me particularly happy is a lot of interaction between the scientists and all the participants in this webinar. So, as I promised, it was long, but I hope it was worth it. So, goodbye, everybody. Take care, stay safe, and see you soon.